welcome everyone to the Silverstein Foundation meeting for January 21st. It is really great to see everybody in person in New York in October, and look forward to seeing all of you again sometime soon. For those of you in the northern part of the United States, I hope the Silverstein Foundation jacket is keeping you warm because it was 11 degrees this morning when I walked my dog, and she was not happy about it. I was less happy about it. But we've got a full schedule today, and we have a guest speaker, Thomas Durkin, who's going to tell us a little bit about their efforts in the GBA1 syndicate. And it might be a possibility for some of the labs that are on here to collaborate with them. So we're always looking to help assist in that collaboration. So Ben, do you want to introduce him?
Sure, happy to. Give you one second here. All right, so this is our pleasure to introduce Professor Thomas Durkin, Director of the EDDU at the Neuro and a lead facilitator of the GCAN network. He's a specialist in patient-derived models and compound screening. Dr. Durkin joins us to outline the GBA1 research tools available to Silverstein Fellows through the GCAN initiative. Take it away, Dr. Durkin.
Great. Thank you, Jonathan. Thank you, Ben. Hello, good morning, everybody, or good afternoon, wherever you may be located today. I'm just going to share my screen. Okay, do you see that okay?
Yes, we can see that.
Nice. All right, you should see the full screen. Yeah,
Yep.
perfect. All right, yeah, so it's nice to meet you all today and actually just present, for a couple of minutes about some of the work we're doing up in Montreal. So I'm working with my colleague Ziv Ganor. So I actually kind of oversee what we call the Neuro's Early Drug Discovery Unit in McGill. And today I'm just going to talk about some of the work we've done over the last few years with Ziv Ganor and kind of through GBA and GCAN and how we're making this kind of iPSC collection, what it is, how you can access it, and if there's other things we do, if you want to work with us then. So I'm really going to focus on more GBA1 Canada. I know probably many of you have met Ziv. So Ziv is actually the director of GBA1 Canada. And over the last few years, it's kind of really taken off across the different sites within McGill and further afield. It's funded through the Weston, through the Silverstein and Van Berken Foundations. And it's built a number of resources that can help people if you want to work on GBA1 and kind of to help accelerate your work. And that's kind of what I'm going to focus on today. So GPA1 Canada, or GCAN for short, we call it. So kind of a unique collaborative model that kind of has been implemented over the last kind of 2, 3 years, but has been really kind of the brainchild of Ziv for years before, kind of to really try to build an integrated approach to take all the data from individuals with GPA1 Parkinson's that you can get imaging, sequencing, pathology, and then start to bring it then over to more kind of models on a dish or even kind of in vivo models, so organoid cell models, animal models, and then to start bringing all of this into big data and to bring in academics, companies to then really focus on both discovery and translating this into something around kind of therapeutics for GBA1 Parkinson's. So what I do is I've actually over the last 10 years about the Neuro and McGill, we've kind of built a group called the Neuro's Early Drug Discovery Unit, or we call it EDDU for short. Some of you may have heard about it, many of you may not, that's fine. We're kind of an integrated group. Everything is focused on human iPSCs at the core, how to make the iPSCs from individuals with Parkinson's and other disorders, then to kind of really do high quality QC, CRISPR editing, and really the guts of what we do is really kind of make them into models that we can go and see kind of help researchers and companies on the first step in the discovery process. You have a target, you have an idea, you know, is this a real idea? Is it testable? We have the models to then do it on a dish. We have the infrastructure, the team, and then we kind of help work with the groups to kind of see, can this go from a target to then potentially therapeutic, and then see, is it actually real and could actually then further go into the clinic. But to do this, we really kind of have built quite a lot of our own iPSCs centered around very specific targets. So, in particular, which is going to be the focus of what I'm going to talk about today, is actually our GBA1 Canada iPSC collection. And just, I'm going to give you an overview. It's about 40 lines. I'm not going to go into all the details. I've given Ben and the group a bunch of material around it, so please access it as well. You can kind of see what we wanted to do is we at the start we knew we wanted to study GBA1, but to do that, you kind of really needed all the tools. So over the last kind of few years, we've really gone and built quite a substantial panel of about 14 iPSCs with individuals with GBA1 defined heterozygous mutations and made the matching isogenic corrections for them. We've also gone and looked at different modifier genes from TMM175 to LRRK2 and other genes as well. So these are all now made. There is one or two more that we're making the isogenics now. But if you're ever kind of looking at the fine mutation like alpha-4p, we have a number of different patient lines matched with isogenics that we are making available, and other users have actually been able to access them as well through our Neuro's Open Biobank. We've also gone and made a number of knockouts. So to kind of see, the gene may be mutated, but what happens when it's not there to help with antibody validation through Lycurus or even just to kind of test to see for specific dark genome. So this is the list of knockout lines you've made, and kind of even gone even one step further, not just to take it from individuals that have this mutation, but to actually work with our own control lines. We have our own male and female line we use extensively, and we've actually gone and knocked in mutations, both homozygous mutations and heterozygous mutations, and kind of the most common ones that people have both requested and are interested in studying. So this is the E36K, N370S, Alpha-4P, and then more recently we've gone and also added mutations in SNCA, TMEM, and so on. So today we've made close to about 40 iPSCs. They are, if it's on the list, it's available in the biobank and people can access it. And I'll talk, in a sec about how you can work with them. But you can actually do a lot. And it's really the goal of it isn't just to have cells sitting in a freezer. We actually want to get it to people. You can use it for a lot of different stuff. Just to give you one or two little examples of it, this is actually dyes that David Vocadlio's group has developed actually out in Simon Fraser. So it's actually now commercially available through BioLegend. So for years we would actually do a lot of our GK's activity in Formog assays to show with these cells, when you make a neuron, you have diminished activity. So we wanted to kind of see, could we have more of an imaging-based approach? And luckily, these dyes were developed with the Fox Foundation. We were able to buy them, and so we've been able to bring them into the group last year. And they've worked really well, actually, with our high-content imaging systems, with the LISO-FQ and the LISO-FIX. So it's just you make your neuron, you add in your dye, and you could then kind of monitor very quickly changes in the activity pretty well. This gives you an example of it. So we kind of wanted to show two lines. We have what we call our isogenic, so it's called 3575, and we have 3575 alpha-4p. So this is a GVA1 Parkinson's line that just had this mutation, and we made 4-week-old dopaminergic neurons. So we would go from iPSC to a dopaminergic progenitor. It takes 3-4 weeks, and then from the progenitor into a neuron. And so once we have the neuron, we would then just add the dye. We would then do high content imaging, and then you can actually quantify changes actually at the activity very quickly within 2 hours. So you can see here, you have your isogenic, you actually have normal or pretty kind of elevated activity. And when the mutation is present, then the activity is greatly diminished. We had other controls that we do add. So if you just do kind of no dye, the control, there's no signal detected. And if you add CBE to just completely diminish it, the activity goes as well. So we did it, but with the live dye, fixed dye, you can actually really kind of across different lines profile GK's activity very well. And so this is tools we've actually used both with many researchers, some companies have come and looked for them, and they're available both. We can actually explain how to do it. We can help you work with them. And, there are ways to then get it going in your lab if you actually have the imaging and want the cells as well. Just to give you an example of so this is actually going to come out in the coming days. It's actually been accepted in Nature Cell Biology. So just to show that these lines, we don't keep them to ourselves. We do make them available. Maria Juan, who's actually a collaborator of mine, actually from Alberta. She does a lot of kind of high content, not high content, more high magnification imaging, really looking at vesicle release. And so she's been able to show with some of these mutant lines that we've been able to provide, there's actually changes that you could actually show ectosomes that would get released containing the synuclein, and this actually was able to then propagate to pathology. So it's in bioRxiv now. It's free to read, but it is actually going to be coming out in Nature Cell Biology probably in the next week or two. It was accepted before the holidays. But just to give an example of what the lines can be used for. And so in a way, we're happy to then provide the lines. We do actually have a pipeline within our group of what people can do. You know, if you've never worked at iPSCs, but you've always wanted to, we do have a whole training program. We actually do videos as well. We know not everyone's based in Montreal. Sometimes people just want to see what we do. So we actually have our own videos on our portal. We have ability if you ever kind of want to make a line, if you know, you think there's a reporter line that would be of great interest. We actually do help people make them. We've made a few as well in the past. And we do all our own QC in-house, and we've kind of got our toolbox for QC to actually make the cells to actually help then, kind of help with the first step of this is the target. Where is it? Is it doing what it's meant to do? What happens when the mutation's there? Is there a phenotype? And now can actually then test different ideas or test therapeutic as well. So as I said, yeah, this is kind of a big part of what we do is outreach and training. So we do have our IPSC seminar series. If people do actually work on IPSCs and they want to actually present, we're more than welcome. It is virtual. We're happy to then have people work on that. We do workshops as well, less frequently now ever since COVID, but we do still kind of offer a chance if a few people do want to come. We do actually have them come work in a lab for a week or two. Some of it's virtual training. We do also a lot of undergraduate training within the McGill community and also even further afield within Montreal. And so this kind of just gives you an idea of some of the stuff we do. And there's many ways we can help you. This is just kind of a overview of some of it. You know, we can help you develop, your own iPSCs, help with disease modeling in a dish, training if that's important to you, exploring how you can even apply new technologies with these iPSC models, and then eventually maybe even testing a candidate therapeutic if you have something. So these are just some of the links. I've given all the information to Jonathan to share, but if you ever want to know more, this is like a very short snapshot of what we do. We have a website, we have a portal, Instagram, LinkedIn. We actually, a lot of the lines we have, they're all registered on the European Biorepository, HPSC Reg, along with the quality control to get it. So you're free to get it there. And we actually have a whole catalog as well that we've shared too. So how the process works is you want certain lines, you identify it, you go to the biobank, and then within the coming months, depending on the two contract offices at the groups, we can then ship you the lines and you're free then to start working with them. So I'll stop there. I know you have a busy schedule ahead of you today, but if you do want to talk more about stem cells, what about we do? Happy to talk. That's my email, and I'll finish up there. And if it's time for questions, I'm happy to take them as well.
Thomas Mulder here. Just a quick question about which line you guys are using to build the knock-in ones and the
So
knockout.
we have our own two lines. We have an AIW22 line and we have QPN. So they're from a healthy control female and a healthy control male. We have talked to the Fox Foundation in the past, about getting their COLF2 line and we could do it all there, but it's all funding dependent. We haven't, we have the reagents and the guides so we could make a third line if need be. But we know that Jax and others have gone and made the knock-ins, so it's maybe a bit of redundancy to go make them. So we have talked to Jax, and the idea is that we'll get some of their knock-ins. We're going to share our knock-ins with them and do kind of cross-validation, probably starting from April, May onwards. So then that'll kind of, you'll hear more about that in due course.
And how easy it is to acquire the lines? It is just an MTA or there is like a purchase process? Like in JAX, the MTA is kind of streamlined. I'm just trying to think through because when it comes to iPSCs, it becomes also the human subject kind of MTA involvement.
We've spent a lot of time trying to streamline this as much as possible. So we do have kind of a point of contact within the biobank. So when you need it, they'll send you all the details. We do need a project plan. What it then goes is we have a tissue data committee that just looks at what is the plan and how are you going to contribute to open science. Like ideally, we're not asking for you to send us all the data right away. It's more that, if you generate data or there's something done with the lines, you could then make it available in the open and then in the next few weeks, or takes a couple, weeks depending on when the committee meets. They'll sign off, and then it'll go to the contracting office, and then that's as quickly we've done in the last 6 months. Made steps to streamline it a lot faster. That it's a very standard template. Often groups would come back asking for a lot of changes. Now it's more like here's the MTA. If you want the cells, please sign it, and you can get it. So it's a lot faster now, but it could take 2, 3 months.
All right. Thank you very much, Thomas, and we gotta move along. We've got a full day today, and we're starting, I guess, in Israel.
Hi,
Hi there.
can I share my presentation?
I can't see, I can see you, but I have the presentation, yeah.
Okay, just a sec.
There he goes. Very good.
Just a brief reminder, Jonathan, how many minutes does each one of us get?
5 to 10, 5 to 10.
And you like those who do 5 and less so those who do 10.
Yeah, we have a big group, so yes, I agree.
Okay.
Okay, so can I start?
Yeah, Shahar, please.
Okay. So hi, my name is Shahar Shani. I'm conducting my postdoctoral in Roy Askar's lab, and today I'll give you a brief update on my work. So as I mentioned in our last talk, we decided to focus on the role of the immune system in GBA1-PD in terms of disease penetrance, age at onset, severity, and progression. Before I start, I'd like to briefly remind you of the aims of this project. The first aim is to use monocyte RNA sequencing data to investigate differences between GBA1 carriers with PD and unaffected GBA1 carriers. The second aim is to perform single-cell RNA sequencing of CSF to identify immune-related differences within the brain environment and to determine whether peripheral immune signals are also reflected in the CNS. Finally, we aim to develop a novel immune-related polygenic risk score specifically for GBA1 carriers with the goal of predicting disease penetrance and association with clinical features. As time is limited today, I will focus on our progress in aim 1. So before presenting our research progress, I'll remind you one of the cohorts we have in the lab, which was established in collaboration with Biogen about 5 years ago. This is a multi-omics cohort of approximately 800 participants, including PD patients, first-degree relatives, and healthy controls, with each group enriched with GBA1 carriers. For this cohort, we have multiple layers of omics data, including DNA-level data, ATAC-seq, methylation sequencing, and RNA-seq from both whole blood and isolated monocytes. In the aim I will present today, we focus on monocyte RNA sequencing data, as monocytes are the key innate immune cells that can infiltrate to the CNS and contribute to PD-related neuroinflammation. So first, we collected whole blood from each participant. We then incubated the white blood cells with CD14 and CD45 antibodies to label monocytes, which allowed us to isolate this population using FACS sorting. You can see this. And finally, the purified monocytes underwent RNA sequencing, generating raw FASTQ files that were used for all the subsequent analysis. A FASTQ file is a standard text-based format for storing raw RNA sequencing data containing both the nucleotide sequence and quality score for each base. Our study cohort initially included RNA sequencing data from 794 monocyte samples. We used the Dragon RNA pipeline by Illumina for secondary analysis to transform raw FASTQ files into high-quality gene expression counts. We then performed quality control filtering, which included excluding sample based on their read depth and excluding and conducting sex verification to remove samples with genotype-phenotype mismatches. On the left, the histogram shows the total RNA sequencing reads per sample, which the average sequencing depth is about 43 million reads. On the right, we have the sex verification QC. By plotting the expression of 4 Y-linked genes against the X-linked gene, we saw 2 very distinct clusters that align with the reported sex of the participants, males in blue and females in red. This step was crucial for identifying and excluding any mismatches to maintain the data integrity of our dataset.
امسك يلا بسرعة جيبوا المفتاح.
I will continue. After completing quality control, 772 samples passed QC and were included in the downstream analysis. This means that only about 2.8% of the participants were excluded due to QC filtering. Here you can see a pie chart showing the distribution of samples across genetic and clinical subgroups. In addition to our quality control, we assess the integrity of our RNA using RIN scores. RIN, RNA integrity number, is a score ranging from 1 to 10 that assesses the quality of isolated RNA, with higher values indicating high quality. As shown in this distribution plot, our cohort exhibits very high RNA quality, with RIN scores clustering between 9 and 10. This indicates that the monocyte's RNA was well preserved and not significantly degraded prior to sequencing. Having this high quality, starting material combined with our QC filtering pipeline ensure that the differences we will observe later are due to biological variation rather than technical artifacts or RNA degradation. To summarize where we are in our study design, we have successfully completed the first two critical stages. We performed the next generation sequencing analysis to transform raw sequences into clean RNA-seq read count matrix. Following that, we excluded the quality control and sample filtering, steps that we just reviewed. Our next phase involves constructing a statistical model that carefully adjusts for relevant covariates, performing differential expression analysis to identify significant differences between genetic and clinical groups, and finally, applying functional enrichment analysis to identify the involved key biological pathways. To identify changes in gene expression, we are conducting 4 main comparative analyses. First, we will perform a broad comparison between PD and non-PD. Next, we focus on the impact of GBA1 status. This includes comparing GBA1 carriers with PD to unaffected GBA1 carriers, as well as comparing GBA1 PD patients to unaffected GBA1 minus, which are non-carriers. And finally, we will examine PD versus non-PD, which are non-carriers of GBA1, GBA1 minus. These are the first results we obtained from the differential expression analysis. They are preliminary, as the model is currently adjusted only for age at enrollment and sex, and does not yet include all the relevant covariates. What you can see here is a volcano plot comparing PD versus non-PD. At this stage, I'd like to highlight that we already observed a clear signal with multiple genes showing differential expression. We will next take a closer look at some of the most interesting points in this plot and evaluate their biological relevance. Of course, we plan to perform the full covariate adjustment, and I hope to present the finalized and fully adjusted results at our next meeting. To ensure robust and biological relevant results, we are developing a comprehensive statistical model. The results shown so far are adjusted for age at enrollment and sex. The final model will also include disease duration, technical covariates such as batch effect and RNA quality metrics, and surrogate variables to account for unmeasured technical variation. This approach will allow us to focus on gene expression differences driven by the diagnosis rather than technical or demographic confounders. I would like to thank all my lab members and our collaborators, and I would like to thank you for listening. Do you have any questions?
Hi, I have a question regarding disease duration as a covariate.
Yes.
Do you intend to use it to regress out the effect of disease duration or to dissect the effect of disease duration to understand it? So to parse, say, your samples and check the transcriptional changes based on disease duration?
No, so I'm planning to put the disease duration as covariate. I will calculate the disease duration according to the demographics data we have, the age at enrollment, and the age of the signals that the symptoms began. and then we will insert it as a covariate in the adjusted model.
So
Okay.
just to clarify, the BEAT protocol recruited people that are newly diagnosed. So they're not medication naive, but they're within 2 years of diagnosis. So the cohort that we have the monocytes on is relatively, there's not a lot of variation in disease duration. They're not people with 10, 15 years of disease. So we
Okay.
won't be able, like, we're not, like, it's actually nice about the the cohort. It's homogeneous with regards to disease duration.
Got you. Thank you.
I have one quick question. Great talk. It's very exciting data, and looking forward to hearing more. As probably you know, our lab has generated monocytes data from GBA1, and we're more than happy to share if it helps you with replication. I know Roy knows about this, but we've generated large number of monocyte. Danielle is going to touch a little bit, but there is a preprint we can share. If you're interested for replication, we're happy to share the raw data.
Thank you.
Yep.
Okay. Any other questions?
All right, we can move on. Thank you, Shahar. Julia, you're up.
Yes, I'll share my screen. Okay. Can you see my screen?
Yes.
Okay. So I guess good evening to some of us, good morning to others. I'm Giulia and I work in Bolzano in Italy at Europe Research, and we mainly focus on secondary mitochondrial dysfunction in the context of Parkinson. So I'll start a bit with of introduction. Okay. So we work with SLP2 protein, which is a mitochondrial protein that is a scaffolding protein that helps in shaping the cardiolipin-rich microdomains within the inner mitochondrial membrane, and basically this facilitates the assembly of the electron transport chain and their function. Moreover, this protein also interacts with different proteins that regulate for mitochondrial dynamics, such as OPA1 and also mitofusin. And we know that by previous work that this protein interacts with Parkin. In samples of Parkinson brain tissue, PD brain tissue, this protein is reduced, and we hypothesize that this reduction leads to the stabilization of protein lipid complexes, and this makes the cells vulnerable to oxidative stress, and so all the events that lead to the death of dopaminergic neurons. And this theory was, let's say, proved by a colleague of ours that works with C. elegans, that basically showed that worms that are mutant for SLP2, when they are exposed to oxidative stress, develop slower. Their neurons are more vulnerable to toxicity induced by alpha-synuclein, and the oxidative stress alters level of phospholipid in the mitochondrial in the membrane, such as cardiolipin and the phosphatidic acid, as you can see here from the staining. And this alteration in these lipids is also accompanied by an altered SLP2 distribution within the mitochondria. So there is the idea that this protein binds to these key lipids, and indeed, here you can see that human SLP2 binds very well cardiolipin and phosphatidic acid. Another interesting thing is that when you have SLP2 knockout swarm, you have a dysregulated mitochondrial unfolded protein response. So we think that SLP2 works like a kind of sensor by binding different species of lipids, in particular phosphatidic acid and cardiolipin, to stimulate the mitochondrial unfolded protein response in the context of oxidative stress. And the previous work in our lab, basically by modulating the SLP2 expression in different PD models, basically demonstrated the beneficial effect of this technique, because in Parkin models, both in vitro and in vivo models, we have our rescue of mitochondrial rescue of mitochondrial morphology and complex 1 activity, and in vivo we also have a rescue of motor function. The same beneficial effects were also seen in alpha-synuclein models, where again we can rescue mitochondrial morphology and reduce the accumulation of phosphoalpha-synuclein at the mitochondria. In vivo, we have a neuroprotection against toxicity induced by alpha-synuclein. So, given the positive results we have on rescue mitochondrial functionality in these two different Parkinson disease models, and given the fact that now we have a lot of data that indicate that genetic variability in all of a series of genes that encode for lipid synthesis and that can influence this pathway are associated with an increased risk of PD, we want to expand the target of this SLP2 modulation to also GBA1, that, as you know, is associated with different alteration in lipid class, including cardiolipin, and moreover, it has been recently and nicely demonstrated that the wild type and the mutant GKs go into the mitochondria and actually are involved in preserving the integrity and the functionality of complex 1. So I will show some results on what we have done so far in this SLP2 modulation approach. So to start, the model we are using are neural progenitor cells. We have, for now, I've been working with a cell line with the L44P mutation derived from a patient. And we want to use progenitors because you can think of them as a compromise because they have present neuronal-like mitochondrial features and they are easier to handle. And we want to develop some high-throughput pipelines to verify some mitochondrial phenotypes, and they are easier to use than fully differentiated neurons. So we generated MPCs from these GBA1 mutant cell lines, and we characterized them for the correct phenotype and checked for some, let's say, basic phenotypes. So we see a reduced enzymatic activity of the GCase enzyme as expected, but we do not see differences in the content of the GCase enzyme. What do we see is an alteration in the autophagy pathway and an accumulation of total alpha-synuclein level, which could be correlated to this blockage of autophagy. Regarding more specific mitochondrial phenotype, what do we see is a reduction of protein that regulate mitochondrial fusion, such as mitofusin and OPA1, which could indicate a possible alteration of the mitochondrial network. And functionality-wise, we see a reduction of ATP content when we force cells to do oxidative phosphorylation, and we see a reduced complex 1 activity. We are checking also other phenotypes such as ROS content, oxygen consumption rate, and morphology. As I was saying, we are trying to exploit this Opera Phoenix high-throughput microscope, and we are trying to set up a bit of different pipelines to check for some imaging-based phenotypes. So by taking inspiration by some already published paper, what we did for now is checking the mitochondrial membrane potential of these cells, and we can see that they are slightly depolarized. That is consistent with an impaired mitochondrial functionality. We also see a slight increase in mitochondrial ROS content. We are also trying to set up a pipeline to measure mitochondrial morphology. Again, there is a nice publication, very recent, published at the end of last year, which we are using as a, let's say, as a starting point. I've been trying for now to adapt this pipeline to our cellular model, and we see that we are able to nicely see the difference between a normal mitochondrial network and a hyper-fermenting mitochondrial network. The next step will be to try and to see if we can detect differences in our patient cell lines versus the control. And regarding interventional strategies that we propose, as you can see from the image here, we want to move on different fronts. But for now, I'll be focusing on this first approach. It is, we have the direct modulation of SLP2. We want to try and do it in two different ways, by mildly overexpressing SLP2 through an antiviral approach, and then a second, more translational approach where we use long non-coding RNA to physiologically increase protein expression without altering the mRNA content. So for now, I will show the result on the approach with the lentivirus. So we set up a different condition for infection to have what we want, so mild physiological overexpression, and we saw that 26 or 24 hours with the virus are sufficient to induce our overexpression, and we checked a potential rescue on the mitochondrial phenotypes that we have seen up to now. So we have nice results both at 6 and 24 hours on the mitochondrial membrane potential. As you can see, the mitochondria get nicely repolarized to a level similar to the control. And we also have hopeful but still ongoing data on the rescue on complex 1 activity. As soon as we finish with the complex 1, we also plan to check the rescue in ATP content and the reduction of oxidative species in the mitochondria. To finish, since for now we just have data on the one cell line with the more severe mutation, we were able to recently acquire in the lab new cell lines. So now we have from Jackson lab, we have cell lines carrying also the milder mutation, the N370S and the risk factor 326K, from which we will generate progenitors and characterize mitochondrial phenotypes also in these mutations and check for the beneficial effect of the SLP2 overexpression. Then we will move as a next step to the more translational approach, the SLP2 long encoding RNA to stimulate SLP2, and we will move also towards the other approaches. First of all, the mini peptides approach, where we plan to use mini peptides derived from Parkin to stabilize the interaction of SLP2 in the mitochondrial membrane, and hopefully also in this case rescue mitochondrial dysfunction. As a last thing, we will also start briefly to generate organoids from this L44P line, which we have already characterized in 2D, to verify these mitochondrial phenotypes, but also more phenotypes that we can get with more differentiation in a 3D model. And with this, I thank my team back in Bolzano, and I thank you for your attention. If you have any question, I'm happy to answer it.
Hi, sorry, I have a question. Maybe a few of them. I was wondering if you're using another mitochondrial or toxin to see mitochondrial dysfunction except then FCCCP. Are you using like antimycin A or oligomycin to see something? Because FCCCP is known to even disrupt other kind of membrane than just mitochondrial membrane. And so I was wondering if you...
Yes, for the MMP assay, I also use antimycin and oligomycin to see if they respond to different kind of, if they hyperpolarize or depolarize. I also use different time points, different concentration, obviously in different assay. Also to induce fragmentation, I used oligomycin and they respond in the same way. I leave them usually overnight with a lower concentration. They survive, but I get the nice fragmented phenotype of the mitochondria. I also try to use rotenone to induce fragmentation and also to check for complex 1 activity. So I try all the possible inhibitors.
Okay, any more questions? Okay, Linda apparently is defending her PhD tomorrow, so Dan is going to step in and give the presentation.
Hi, thank you. Yes, indeed. Yes, apologies for Linda not being there. She's undoubtedly very busy at the moment. So our research focuses mainly on the role of sphingolipids in GBA-PD, and a bit more specifically, we want to investigate how GBA dysregulation leads to altered GSL metabolism in GBA-PD related to IPD, but also to related to Gaucher disease. I will go fairly quickly through the introduction and focus on the latest results, because what we have been doing in our lab in my fellowship when I was a fellow is look at different models of GBA-PD and see what the GSL expression there is, so glycosphingolipid expression, but also in collaboration with Mina Wright and relate that to PD gene expression and then specifically glycosphingolipid pathways. I will not go over all these results now. As I mentioned, we have had many collaborations that indicated multiple changes in glycosphingolipid levels in the plasma, human plasma initially, but also in different models. And what I would like to focus on is our latest sort of validation study or continuation, in which we increased the sample size of the human plasmas to get a clearer picture of this. This is in collaboration with from plasma samples from Columbia University with Dr. Ray Alcalay, and we are now collaborating with Steen Lab and with Velling Lab to share these plasma samples so that we can do sort of a multi-omic analysis, but they will also talk a little bit more about this. So where this all came from, we analyzed GBA PD plasma coming from UCL, from Anthony Shapiro. And what we found initially is that indeed GBA carriers cause an increase in total GSL levels, and that we see that many GSLs are upregulated. We saw, however, that there was quite a strong gender effect there. If we look closer to the biosynthesis pathway, we see that mainly certain parts of the pathway are affected, and we see that gender effect is also very prominent there. So we wanted to expand this. We did. So we analyzed about 150 samples, both males and females, stratified, in which we analyzed the controls, IPD, GBA, and GBA-PD. But here we had the opportunity to also look into the specific variations of GBA mutations. As mentioned, this is then a collaborational effort at the moment, in which we look at the glycosphingolipids, Steen Lab looks at the proteomics, which we'll hear more about, and Fanning Lab goes more into the fatty acid lipidomics, so that we can do this multi-omic analysis. Back to my results, I analyzed these plasma samples, and what we found basically is very much in line with the initial results, is that we saw that in GBA-PD, the glycosphingolipids generally are heavily upregulated. That was basically for all individual GSLs the case. When we look at the females, we saw that this effect was very much there indeed, maybe even stronger. But interestingly here is because we increased the sample size and we were able to look at the individual GSLs, we saw an increase in IPD as well, which we did not observe earlier. When we look at the males, however, we see, if we look at the individual levels, also an increase, be it milder, of total GSL levels. But here we see a slight decrease in IPD. So that's quite the opposite effect and quite a strong gender effect again. So we see that most of the GSLs are upregulated in females in GBA-PD, not so much individually in the males. Together they are, but there's a downregulation then in the males as in IPD. Now, because we had the chance to look at the individual mutations as well, we saw that this might be most strongly upregulated by specifically N370S. Now we see that everything goes up a bit, but that the milder mutations might also cause milder upregulation in glycosphingolipids. Again, this is most prominent in the females here. So that, as a summary, we saw that the GSLs are upregulated in GBA and also GBA-PD in males and females. 370S might be the strongest. But interestingly, we saw a differential effect in IPD. Now, as a very initial and preliminary hypothesis, it looks like from our data that because we saw quite a strong upregulation in specifically males, but those were non-manifesting carriers. So what it seems like is that this is very much GSLs are upregulated in males. However, the PD slightly brings this down. So we have an upregulation GBA PD in males. But in females, it seems stronger because also PD seems to upregulate this. This is very much in line with our first analyses, and this is what we are now investigating further. So the following steps are basically taking this data and comparing this with all the previous data sets of PD that we have acquired in the lab as well, and throwing it together into a very large data set to validate this and to see if different aspects, we're also looking into different clinical aspects play a role there to explain these findings. And as mentioned, we're looking into integrating this data with the proteomic and fatty acid analysis in the other labs. And next steps, and especially for Linda, this will be how to relate this to the RNA sequencing and GBA PD with collaboration of the Writer lab, and of course the integration write an integration paper of basically the model organisms and these analyses that we found there. So that was very shortly an update of what we have been doing, and we're very happy to take some questions. Thank you.
All right. No questions. We can move on to Alberto. Thanks, Dan.
I have a question.
Okay.
Just a quick one. Dan, do you have the ApoE status for these particular patients, and is there any effect with respect to males and females?
I do not have those at the minute. We are acquiring some more clinical data from these samples, but we don't have that yet. And this is something that we can indeed look into what is available. I'm not entirely sure if APE status is in that. Would be very interesting indeed.
Dan, and excellent presentation. We know that in more commonly tested lipids, there's a huge difference between men and women. So I think it's very hard to interpret the results. I think we can try, but it's not surprising because triglycerides, the LDL, like the very abundant lipids in blood are completely different.
Yeah. Yes. Thanks. Thank you.
Okay, Alberto.
Hi, let me share my screen. Can you see my screen?
Yes.
So, hi everyone. Would like to give a brief update on the project we've been carrying on with the group of Enza Maria Valente in Pavia, Italy. So just a brief reminder of the aims of the project. Basically, we are interested in investigating these 4 factors you can see in the cartoon, so GBA1 variants, and how they interact with sex, and specifically with sex hormonal profiles in both females and males patients, and GBA1 carriers without PD, but also with the peripheral mitochondrial damage and oxidative stress, and how these factors interact with the homeostasis of peripheral alpha-synuclein, so the homeostasis between the monomeric form and the aggregated form, and then how these factors interact together to explain different trajectories, clinical trajectories in PD patients, and the risk of conversion in GBA1 asymptomatic carriers. So the outline of the project was to enroll 14 non-GBA PD patients, 40 GBA PD, 25 GBA asymptomatic carriers, and 25 idiopathic PD, and then to conduct a clinical evaluation at baseline blood sampling following a series of biochemical analysis focused on mitochondrial markers, sex hormonal profile, and as well GCase activity and peripheral alpha-synuclein in PBMC evaluation. And focusing on the AIM-1, I want to share a brief outlook of the protocol we are evaluating our patients. So we are conducting a deep clinical phenotyping, including motor domain, functional activities, and fatigue, as well as physical activity, quality of life, nutrition, but also a battery of exams of non-motor function, including sleepiness, pain, olfaction, etc., as well as a complete reproductive, andrological, and gynecological history in all participants, as well as the menopause rating scale, which assesses the main symptoms which are associated in menopause in women patients. We are also conducting a complete battery of neuropsychological evaluation, including, of course, global cognition and all neuropsychological domain, visuospatial, language, memory, and executive function in all participants, as well as the psychiatric domain, including the impulsive, compulsive disorders, depression, anxiety, etc. And going on to the AIM-2, so the biochemical analysis, we are adopting these protocols. So specifically for GCase activity, we are using an in-house already validated protocol using fluorometry and the 4-MUG as a fluorogenic substrate for GCase activity in PBMCs. And then also alpha-synuclein. We are evaluating alpha-synuclein levels in PBMC in its monomeric form using an ELISA kit from Legend Biomax, and also the aggregated form. I put the aggregated form in red because we have several technical problems starting from the start of 2025 within a parallel project with this kit. So in particular, the blank of the standard curve was abnormally higher in the last experiment we did in the start of 2025. Next, the kit was withdrawn from the market, and it has been just introduced to the market in the last month. So we are going to do in the next month the first experiment to see if the results are reproducible compared to our old experiments. And if you have any suggestion on other kits, ELISA kits that are useful for the evaluation of aggregated alpha-synuclein on PBMC, we are very happy to take any suggestion, of course. For the sex hormones, we are collaborating with our diagnostic lab, which is using electrochemiluminescence immunoassay for this battery of hormone in plasma. And we are still defining the protocols for the evaluation of mitochondrial cell-free DNA in plasma, redox state, as well as the mitochondrial extracellular vesicles. So this is the partial clinical demographic features of the patients we have enrolled until now. So we have currently 25 GBA PD patients, 19 idiopathic PD, 13 GBA variant carriers asymptomatic, and 27 healthy controls without GBA1 variants. And for many of these patients, around 70 or 80%, we have already in our biobank retrospective plasma collected that we can use also for longitudinal biochemical analysis in the next months. And also for many patients, we have retrospective clinical data in our REDCap. So we can plan to do correlations between the biochemical analysis and the longitudinal clinical data. So this was the original project timeline. We have now enrolled 84 patients on a total of 130. So we are about the 60% of the enrollment initially planned. And as I said before, the GCase, monomeric alpha-synuclein sex hormone biochemical analysis are already ongoing, while we are still to start with the mitochondrial analysis. And about the retrospective data, we have a total of 58 participants on 84 with full retrospective clinical data and 60 participants with retrospective plasma available in our biobank. So this is all for today, and I would like to thank Ensa Maria, of course, and all my team, which is collaborating in these projects. Thank you. I'm happy to take any questions, of course.
Any questions? Okay, we can move on to Noemi.
Yep. I share my screen. Okay. Can you see my screen?
Yes.
Okay, perfect. So hello, everyone. So today I will do a quick update on the project that is going on in our lab in Luxembourg in Jens Schwamborn team. So which is based basically in the stratification of GBA, PD organoids to try to identify different profile and adapt personalized therapeutic strategy for that patient. So as you know, and quick introduction, the one-size-fits-all therapy for the GBA PD patient and globally for a PD is not working since now. So because we are just considering the mutation in itself without taking the personal genetic background of the patient and even maybe some mechanisms that are more involved in some patient than others. And so the idea here is to try to identify different subgroup of GBA PD patient and adapt the therapeutic strategy on these subgroups. So to do that, the different objective I here explain. So the idea is to generate different midbrain organoids, healthy control, GBA PD, and as well GBA asymptomatic midbrain organoid for patient. And first of all, we will do in a first step a phenotyping analysis. And then we will combine this phenotypic analysis with a genotyping analysis to then identify this profile and subprofile and start to adapt the treatment for each group. So today I will focus more in the first part, which is the phenotyping analysis. So as a test phase to start to have some phenotyping characterization of our midbrain organoid, I started to use 3 controls lines versus 4 GBA PD lines, knowing that normally we, for the entire project, we manage and we plan to use 6 GBA PD lines and 2 asymptomatic lines. And so we culture this midbrain organoid for 30 days and then analyze them. So as a quick reminder, we start midbrain organoid generation from NESCs that are neuronal stem cells. And we have 2 different approaches for imaging. We embed them in gel tracks to create a kind of matrix around the organoid and lead them to create the projection in this matrix, and so we can observe better the morphology of the neurons. And for other kind of analysis, such as qPCR, Western blot, we will use non-embedded organoids that are remaining static in the plate. So this is how they are looking like. So in the upper lane, you can see the non-embedded organoid, and in the down in the other lane, you can see the embedded organoid that are showing nice projection in the gel tracks. So as a first thing that we checked, it's the consistency of our organoid size between our different cell lines, as it could be that the size could be related to some cell death. Sometimes is what we noticed in the team. So here we use the plate reader with an objective microscope to check with a not super great resolution, not like a confocal, but it's enough to normally check the surface and the area of the organoid and to establish the size. And so as you can see, there is a variability of the size between our cell lines. However, every dot is an organoid generated, and mostly every organoid generated has a consistent size in between each cell line. And then another thing that we checked is how is our TH protein level in our midbrain organoid, and to answer to that we did Western blot, and so we checked TH level as a protein corresponding to the dopaminergic neuron population. And as you can see here, globally in our GBA-PD, we can see a decrease of the TH protein level compared to the wild type, and this decreased is not seen in the neuronal, total neuronal population that is represented by TUJ1 protein. So here we can see that in the GBA-PD, we have a loss of dopaminergic protein, let's say, that is specific for that population. And when we split, let's say, our GBA-PD line in the for each cell line, you can see that globally we still see this decrease of TH protein level, but some cell lines are showing a more severe, let's say, loss of that protein compared to other. To validate that data, we checked in another site by immunofluorescence the dopaminergic neuron population, and to do that, we did immunostaining on organoid section that are having a thickness of 60 microns. And as you can see here in this representative picture, we can see on the right side that the GBA-PD lines are showing less dopaminergic neuron that are stained by TH, and as well a non-ramified, let's say, network compared to the wild type. And here are the quantification of the staining. So globally, we can see this decrease of dopaminergic neuron population in the all GBA group. And when we split the cell line again, we can see that this decrease of the dopaminergic neuron population is variable between the cell lines, but it's still giving a tendency to the decrease mostly. And when we check the functionality of the dopaminergic neuron by doing a dopamine ELISA, where basically we are dosing the dopamine that is released in the medium by this dopaminergic neuron, we can see that we have a tendency to the decrease of the dopamine in the medium of GBA-PD organoids. So it's a quick overview of what we have in ongoing. So for now, we are checking that we see a decrease of the dopaminergic neuron population, and that we have a loss of dopamine as well, a release in that sample. And another thing that we would like to see in the neuronal, phenotyping characterization is the alpha-synuclein aggregation and the dopaminergic neuron fragmentation that is mostly, let's say, parallel to the dopaminergic neuron loss. And then we will go more deeply in the characterization by looking organelles function, so lysosomal function, autophagy function, and mitochondrial function, and probably ER function too, which is quite underlined in this late paper in GBA patient as well. So I would like to thank you for your attention, and I'm open for question. Thank you.
Hey, great talk. I was just wondering whether your TH loss, do you think you can predict that from organoid size? Like, are the organoids that are smaller, are they also the ones that have the most TH loss?
It's a great question. The idea could be that. The problem is that we see a variability in size even in our wild type organoids. And sometimes the smallest one are having a biggest TH population. So we don't see really in a systematic point of view this correlation between the size and the differentiation of dopaminergic neuron population. For now.
I have one question. So do you think that there is less dopamine neurons or there is less differentiation of dopamine neurons from the neurons?
It's a good question. There are papers, and even within our team, we describe that in GBA organoids, we can see probably an issue in the differentiation process. So indeed, I started to do the experiment by looking only one time point, that is the day 30 post-differentiation of the midbrain organoids. However, I wanted to introduce, let's say, early time points, where usually we can still see progenitors of the dopaminergic neuron. So the idea is to really quantify as well the dopaminergic progenitors population to see if at the end what we have, it's probably a mixed situation, I would say, but we have to confirm that indeed.
Thank you.
Are you going to quantify also the monomeric form, so the functional form of alpha-synuclein? Because it would be interesting to see if the TH dopaminergic neurons loss was some way correlated to the levels of monomeric functional protein.
It's a good question. The monomeric form, so you mean the total one, right?
Yeah, I mean the monomeric one, not aggregated, not phosphorylated, the one that should be functional.
Yeah, so I suppose that to answer to that, alpha-synuclein, usually it's located in synapses when the neurons are mature, right? So it would be interesting to answer to that question if we have an issue in the location of the synuclein in the synapses, or because there is some issue in the synapses, like processes and function of the dopaminergic neuron, the protein can't do what physiologically does. So it's a question that we can try to answer by staining, I would say, mostly.
Thank you.
Thank you.
Sorry, Noemi, a question.
Yeah.
Did you do some functionality test to see if there are differences in this, like electrophysiology, to see if there are differences between patient and control?
I haven't done by myself functionality. I like MEA or all this stuff. I know that in the team we are doing that. I don't know if Jens is still here, but maybe he could add some details about that. For the, you mean in the functionality of the neuron itself? What I know is more about the mitochondria. I know that we see mitochondrial issue in this cell line generally, but we haven't split the cell line by itself to see one by one their metabolic, like mitochondrial profile, and would be great to do the same in MEA, for example. Yeah.
Thank you.
Yeah, I'm still here. By now also I'm parking, so I can answer to it. Yes, we have done multi-electrode arrays on the GBA lines before, and well, indeed, you see a reduction in the activity, particularly like also in the response to quinpirole. Not sure how direct that is. It might be an indirect effect because you have a massive loss of dopaminergic neurons. So it's not a big surprise that you see an impact on activity. And maybe to the question before, like there was basically a question for looking for monomeric alpha-synuclein, and we totally do that just by Western blotting for alpha-synuclein itself.
Okay, thank you.
You're welcome.
Thank you, Jens.
Pleasure.
Okay, Hannah Clark.
Hello. Can you see my screen?
Yes, we can.
There you go. Great. So nice to see you all again. So this is just a short update on what's been going on in our lab. I'm in Sonia Gandhi's lab over at the Crick Institute. So just a brief reminder of what my project was. Ooh, sorry. Can you guys still see that? It looks a bit weird on mine.
Yeah.
Okay, cool. So my project was trying to look at lysosomal niches and how that in vitro models and how that's going to affect aggregation of alpha-synuclein. So I've got three aims of this. So the first aim is to try and establish a midbrain platform from iPSCs and then use that to have a look at aggregation. And then my third aim is slightly separate. So from that, we're going to try and see if we can predict therapy and predict progression based on lysosomal phenotypes in a dish. So first of all, focusing on the first aim, most of my time at the moment has been using this time to curate the IPS cohort. So initially in-house, we had these one male and two female lines and an isogenic control, which have been phenotyped by a previous PhD student. So I'm trying to expand this up. So I've gained a fair few more lines for M370S, and I have ordered in a bunch more lines for E326K. So the idea here is that now I'll have an IPS cohort from M370S and also an E326K cohort. And then hopefully, eventually, once they all arrive, I can get them banked and expanded up and QC'd and then start performing some of the experiments. So what we want to do is lysosomal IP, do lipidomics, proteomics, and then see how this affects aggregation in a dish. So first of all, doing FRET, and then maybe also looking at biochemical assays of aggregation. So at the moment, the progress has been ordering them all in, making sure that we've got a good bank of lines. But unfortunately, they're not here yet. So when they get here, we'll get working on that. And then for aim 3, with the prediction of lysosomal subtypes, for this, we've got a cohort called QMAP PD, and this is a cohort of a bunch of different Parkinson's disease patients, all with various different mutations. And included in that are 10 patients with GBA-PD. And they're going through lots of deep phenotyping, so motor phenotypes, non-motor phenotypes, and then lots of MRIs, both structural and function. And then we've collected their blood and their fibroblasts from this. And the idea for this part of the project is to take the fibroblasts, culture them in the dish, and perform transdifferentiation straight to cortical neurons, and then perform high content imaging, looking at the nucleus, the mitochondria, and the lysosomes, and then try and train a classifier to stratify PD patients with GBA mutations from controls. So at the moment, we have now got all 10 of our patient lines in-house and banked, and the fibroblast cultures are generated, which is great. And I have now ordered in all the bunch of controls, which are age and sex matched. So we're making good progress here. This is just to show that we've managed to age match and sex match our controls. So what we've done so far is we've optimized the direct conversion protocol so we can make good neurons from these. So this is neurons in the dish that are MAP2 positive, and they all seem to fire, which is quite nice, and they have like very functional networks, which is great. And interestingly, we found that if we do the multiplex imaging from fibroblasts, we see different phenotypes from in the fibroblasts and in the neurons. So this may be something to look at, see if we can train our model from fibroblasts and then train our model from neurons and see whether the models predict different phenotypes. So we're making good progress on the model development at the moment, and this is in collaboration with the bioinformatician and mathematician in the lab. And we were planning to collect from fibroblasts, bright field images, this multiplex images of the mitochondria, the lysosomes, and collect this at 2 different time points, just to make sure if passage number and the age of the fibroblasts in the dish will matter. And then we're going to perform transdifferentiation and collect from the neurons, bright fields, and multiplex images, as well as transcriptomics, GCase activity, and proteomics. And then we're going to plug all of this into the models and then also treat the cells and then see if the treatments, see how the models can predict cells that have been treated versus cells that haven't been treated. So the classifiers at the moment, we've got a separate cohort of sporadic PD where you have fast progressors and slow progressors, and the models have been generated and we can successfully remove dead non-neuronal cells just from the images and stratify age and sex, and have created a alpha-synuclein fingerprint based on these tabular data. And these models are doing quite well. So we've got 98% accuracy for the removal of dead and non-neuronal cells from the dish. And the stratification is working up to about 73% at the moment, which is pretty good. And this is for the whole well, and it individually assigns each cell in the well, whether that is from a patient or not from, or if it has like SNCA phenotypes or if it doesn't have SNCA phenotypes. So the idea is to do this, use these models and try them out on our GBA lines and see whether we can do the same stuff from the fibroblasts and from the cortical neurons and create a GBA fingerprint. And then hopefully we can also create some new models to train from the image data and then incorporate all of this extra complexity of data into the models. So the GCase activity, the omics, and see if that helps the prediction. Then also, because we see these different phenotypes in the fibroblasts and in the neurons, see if we can use the fibroblast to predict the neuron. And then can we actually predict whether the cell is from the patients or from controls by just looking at bright field images, which is something that would be very good to look at. So over the next 3 months, this is the plans. I'm going to start very soon, expanding up the fibroblasts and the iPSC when they all arrive, perform some quality control, then start on the high content imaging of the fibroblasts, which should be pretty fast, perform the transdifferentiation, make sure they are making good neurons, and then start doing the multiplex on the neurons and start plugging this into the models. So I reckon within the next 3 months, this should be where we're at. So just thank you for listening. And these are the 2 people that have done a lot of this background work already. Cheers.
Hannah, can I ask, it's beautiful work and really nice plans. In light of Thomas Durkin's presentation this morning, would there be an advantage to using those lines with the isogenic controls to try and overcome some of the human-to-human variability?
Yeah, I think it'll be great. I'm going to drop him some emails as soon as we're off of this call. I think today, I think it'll be really good, especially because at the moment with my E326K lines, I'm struggling to get both male and female, so it'll be quite good to have a look at what's available and see if I can get it a little bit more sex matched as well.
Sounds good.
Any additional questions?
Yeah, maybe Hannah, can I ask you, did you say you see different phenotypes in the fibroblasts as compared to neurons? Is this stronger effects in neurons, or what is the difference?
Yeah, so this was work done on the MSA lines that we have in the lab. And at the fibroblast stage, we don't see any changes in mitochondrial phenotypes. So we've got just TMRM intensity. At the fibroblast, we don't see any effects. But then when we use transdifferentiation protocol and age out the neurons, then we start to see that the MSA mitochondria are more depolarized. So it suggests that this MSA and this very limited group that we've tried it on so far, we can start to peel apart different phenotypes based on the cell type, which I think will be really crucial for the development of the models.
Sure. And the SNCA fingerprint, was this from IPD or SNCA mutant patients? I didn't catch that.
So yeah, that was all sporadic PD. So it was patients which were stratified by a clinician to be either fast progressing or slow progressing. And it came from the PPMI cohort.
Okay. Thank you. A last question, maybe. What type of treatments are you planning to use?
So at the moment, mainly thinking Ambroxol, and we've got a GLP-1 agonist in the lab at the moment. But very open to collaboration. And if anybody's got a drug of interest that they want to chuck on the cells, please let me know and we can try it out.
Okay, great. Thank you very much.
Thank you.
Okay, Camille, you're up.
Try to share my screen. Doesn't work. Okay. Can you see it?
Yes.
Okay, good. Sorry. Okay. Good evening and good morning, everyone. So I will start by a smaller introduction. So we are interested to look at some PD risk genes, CTSB and TMEM175. So on the left, CTSB can cleave the saposin C to make it mature, which is required to help to activate the GCase. And a decrease of the CTSB, the paraminergic neurons, have shown a decrease in GCase activity. For TMEM175, what has been shown recently is that it's a unique channel, so either proton or potassium, and a decrease of TMEM175 can also lead to a decrease of GCase activity. So we were interested to work on those PD risk gene. And also, so for GBA, as you know, like for the PD risk variant, it's also impaired the GCase trafficking to the lysosome and also to decrease of GCase activity. And the unfolding of GCase leads to an accumulation, and which leads to ER stress. So the idea is that we want to use a chaperone that will help to restore the trafficking from the ER to the lysosome, and an activator to increase the intrinsic GCase activity to rescue the GCase trafficking and activity and improve PD-related phenotype, either in GBA1 variants or in CTSB and TMEM cell lines. So as Thomas Durkin present you, at McGill University, they made those patient lines. So GBA variant carrying L44P and 370S, E32K, and also a French-Canadian variant, which is W370AG. So we want to use this line to make dopaminergic neurons, but also we would like to decrease the level of or increase it using the CRISPR interference and activation mechanism to induce or increase CTSB TMEM175 in GBA. So then we would like to treat with either the Ambroxol, so the chaperone alone, or the LTI alone or combined, to see the effect on the GCase trafficking and activity, the lysosomal function and composition, and also the PD targeted assay related to mitochondria, lysosome, or alpha-synuclein accumulation. So first, I've been working on making the CRISPR A and I line. So to give you a brief introduction, so the iPSCs contain a CRISPR-A or CRISPR-I that is TMP inducible. So we inject lentivirus containing the guide RNA and select them with puromycin. And then using the NGN2 and inducible transcription factor, we will force the differentiation into neurons, which is quicker than the other model. And then by adding the TMP, we can generate the CRISPR-E or the CRISPR-I of our gene of interest. So here I show you the result on the iPSC CRISPR-A. So I look at the relative expression compared to the control, which is non-targeting small guide RNA. And you can see for CTSB, GBA, and TMEM175, there is an increase of the relative expression in the CRISPR-A. And for the CRISPR-I, also in the iPSCs, you can see a relative decrease of CTSB, GBA, and TMEM175 compared to the control mock. So then we wanted to validate that the neurons can have a proper differentiation. So by looking at MAP2 neurons, which are in white here, and TH marker, which is in green here. So for the control, for GBA1, CTSB and TMEM175, in this case is for the CRISPR-I line. And we calculate the percentage of the MAP2 neurons and the percentage of TH neurons among the MAP2 neurons, and we see a quite good differentiation. So this has to be validated, but it's a good start. So then we wanted to validate if in this neuron, the level and the colocalization of GBA1 with LAM2. So you can see in green, it's GBA1 marker. In magenta, it's LAM2. In white, it's MAP2. And you can see that in our CRISPR-E, we lost the labeling of GBA1. And we have some expression of GBA1 in CTSB and TMEM175 at the protein level in dopaminergic neurons. So the next step will be to analyze the colocalization between GCase and LAM2. So to finish, to validate this line, we look at the activity of GCase using the LISO-FQ, which has been generated by this group to be specific to the lysosome. And when the GCase is active, it will cleave the quencher, which will emit the fluorescence of the product that you can see in the control. So we look at the LISO-FQ intensity. We use the CB to inhibit the GCase. So you see that there is no activation. And we compare with GBA1 CRISPR-I. So we have a clear decrease, it seems, because it has to be confirmed. But for the first try, it seems to clearly decrease the GBA1 activity. We saw also a decrease for CTSB CRISPR-I dopaminergic neurons, which are 4 weeks old. And there is a slight decrease of the GCase activity in TMEM175 CRISPR-I neurons. So the next step is to validate the CRISPR-A and to finish to quantify the GCase trafficking and to look at the mitochondrial phenotype, lysosomal phenotype, and phosphocene labeling or alpha-cene accumulation. And as those cell lines have a decrease of decays, it could be interesting to use them to try to determine the dose response of the ambroxol, LTI, and the combination of two. And then we will be able to move with the patient line. So I thank you for your attention, and I thank everyone and my colleagues for this project.
Okay, I think we're going to take a 10-minute bio break. So please come back on time so we can finish up the rest of them. Some great presentations today. I'm really happy for the work you guys are doing. Thank you so much. We will be reconvening at 12:40. Okay, thanks. Okay. People are hopefully getting back to their desks. Sinead, I think you're up. Well, let's give it 30 more seconds for people to get to their desks.
Basically, the entire group dialed in like 2 minutes late.
Okay.
Got done 10:58.
Looks good.
Everyone see my slides and hear me?
I can see your slides.
Hear you remotely, yeah.
Okay. So good afternoon, everybody, or good evening. My name is Sinead. I'm with the STEAM Lab, and here's a brief update regarding our project.
Sinead, can you up your volume of your microphone?
Yes, let me see. I can also just use. Sorry, can you hear me better now? Okay. Sorry, I just changed over. So here's our update of this project. So previously in the other meeting, I had shown that we have 3 main research objectives. And today I'll be focusing on objective number 1, which is the proteomic analysis of GBA PD. So really briefly, this is the overview of this first objective. Using both brain samples and plasma samples from Columbia University, we'll be carrying out mass spec-based proteomics in order to identify any potential proteins or pathways of interest in GBA PD, but also within the different GBA mutation groups. Also compare them within each other and also to other synucleinopathies. So today I'm going to be focusing on some preliminary data. This has yet to be validated for the plasma proteomics for the GBA PD neat plasma. And I want to thank Dan for sending us these 150 plasma samples in quick succession. So for this, for our overall plasma proteomics pipeline in the STEAM Lab, we have 2 different methods of sample preparation, both neat plasma and depletion. The neat plasma is using just the raw undepleted blood plasma for protein analysis, while the depleted plasma is using an acid precipitation to deplete the more abundant classical plasma proteins for more potentially disease-relevant proteins. So for these samples, we received 50 controls, 51 IPD, and 48 GBA PD samples. And within this, the 3 mutations, or 3 of the mutations were represented. For L44P, we had 8 samples. For E326K, we had 18 samples. And for the mutation N370S, we had 22 samples. So today, I'm going to be talking about our neat plasma analysis. For this, we only need to use 1 microliter of neat plasma. Because we have 149 samples, they were split over 2 96 well plates, and everything is automated in this pipeline using the Okatron Flex. They're then ran on our mass spec, our timsTOF HT, with the EvoSep system at 100 SPD per day in DIA mode, using a previously generated human plasma proteome library from the STEAM Lab. So one thing that has been really helpful and well established in the STEAM Lab is this fully automated system that we are able to do high throughput plasma proteomics. It has limited human error and increases our ability to analyze hundreds, if not thousands, of plasma proteomic plasma samples. So first of all, we identified from just 1 microliter of plasma 594 proteins on average per sample. And with this automated system, we see a lot of consistency and reproducibility across the different groups also. On average, we saw 595 proteins in the controls, 597 proteins in our IPD group, and 589 proteins pretty consistently identified across the GBA-PD group. So the first thing I look for is the overall quality of the data. What we did initially observe is a batch effect. As I mentioned before, they were run on 2 different 96-well plates. So no outliers were removed because the protein counts were very consistent. When we log transformed and tick normalized the data, we also only kept proteins identified in more than half of all samples, but there was a clear separation based on the 2 plates that were run using PCA. So this was corrected for using the combat to correct, and now we can see there's a bit more variability and there's no clustering according to plate. So the first thing I did was looked at the overall proteome differences in the plasma between the IPD patients and the controls. We identified 15 significantly downregulated and 6 significantly upregulated proteins, including proteins involved in immune and immunity. For then the GBA-PD samples, we identified even more significantly dysregulated proteins, with 71 downregulated proteins and 7 upregulated proteins. A lot of these involved immunoglobulins, and also some complement proteins were dysregulated in this group. I also looked at, well, what was the overlap? We found that there was 2 upregulated and 8 downregulated proteins that were shared between the 2 PD groups, meaning that irrespective of if it was GBA associated or just idiopathic PD, these proteins were dysregulated in the plasma for both sets of patients. I also then looked at GBA PD patients versus the IPD patients. 7 proteins were significantly upregulated, with 43 significantly downregulated proteins. Some of these involved epigenetic regulators also. Overall, when I looked at what was consistently found in GBA-PD patients versus controls and IPD, 26 proteins were significantly downregulated across GBA versus PD and controls. These involved several different immunoglobulins and also several of the classical complement pathway proteins were consistently downregulated in GBA-PD versus IPD and also controls. When we look then at the biological processes, this is shown with complement activation being the highest process that is enriched. Next, I looked at each of the mutations themselves. As mentioned before, for the L44P group, we only had 8 patients. However, we identified 10 significantly upregulated and 2 significantly downregulated proteins. One of the upregulated proteins being FCGBP, which is involved in immune defense. For patients with E326K mutation, we observed the highest number of proteomic differences in the plasma versus the controls, with 18 upregulated and 139 significantly downregulated proteins. Some proteins involved immunoglobulins, the complement proteins again, some serpine proteins, and lysosomal proteins. And then we also looked at N370S versus the controls. We identified 16 upregulated and 22 significantly downregulated proteins. Other proteins included IGFBP2, which is an insulin growth factor, immunoglobulins, complement proteins, and also serpine proteins again. When I looked then at the biological processes for L44P, there was no process enriched. For the E326K, as I mentioned before, it was mainly complement and immunity. And this was the same then for patients with N370S. When then I looked at each mutation versus IPD, we found 8 significant upregulated proteins for L44P patients and 6 downregulated proteins, once again identifying the FCGBP, and VICAM1 was also identified versus IPD and controls. Once again, for the E326K, we saw an increase of 120 significantly downregulated proteins, so the most out of the mutations, And once again, these involved immunoglobulins, complement proteins, and serpine proteins. And for N370S, we identified 5 upregulated and 19 downregulated proteins, once again encompassing a lot of the immunoglobulins and serpine proteins. Lastly, I also then compared each group against one another, so each of the mutations. For L44P versus E326K, there was a huge upregulation of 65 significant upregulated proteins and 9 significantly downregulated proteins. A lot of the upregulated proteins included complement in this group now versus the other mutation, and anticoagulants, and a downregulated protein like MLLT3. When L44P patients were also compared to N370, I saw an upregulation of 18 significantly upregulated and 11 significantly downregulated proteins. And once again, the MLLT3 was found. And then when I compare E326 versus N370, we saw a downregulation of 80 proteins, upregulation of 16, and a downregulation of a lot of the apolipoproteins when we compare these two mutation groups like clusterin, APOF, APOM, and APOC1. So overall, what we found was that patients with the GBA E326 showed the highest number of proteomic differences in their plasma, the highest number of dysregulated proteins. Overall, we identified that there was 18 proteins that were consistently downregulated in these patients across all different comparisons. These are proteins involved in coagulation, the complement proteins as shown here, and some immunoglobulin light chains. When we then looked at what biological processes are enriched in these 18 proteins, we saw once again a lot of anticoagulation, fibrinolysis, and some inflammatory response proteins. So for the next steps, as I mentioned previously, a lot of the proteins that we have found need to be validated. We need to dig deep into this data set and look for any biological relevance or biomarker capability. As shown before or will be talked about, 2 other groups are looking at these samples, and we would like to integrate the lipidomic analysis of these patients also and validate in a second cohort of plasma samples. If possible, increase the number of, for some mutations like the L44P, or even if we were able to obtain GBA positive patients without clinical symptoms, that would add as an additional control group. One thing that we haven't looked at yet is potential sex differences. Is there differences between males and females, which I think is also going to be very interesting. And the next steps really are then to start with our depleted plasma proteomic pipeline. For this, we now want to see, can we identify alpha-synuclein in particular in the samples? And are we seeing some of the disease or the proteins we identify in the neat plasma? So thank you, and let me know if there's any questions.
I'm wondering, that was a nice presentation. I'm wondering if the type or strength of the immune signal that you see in GBA-PD is somehow fundamentally different from the immune signal that's seen when you just compare PD to controls without PD.
Yeah, totally. I think what we're seeing is a very high complement pathway difference. However, it is different complement proteins that are coming up in the different mutations and with PD also.
I don't know if it's helpful to you, but we just published glucosylsphingosine levels as measured in Merck from this cohort.
Okay.
And if that would be something that you would like to add into your algorithm, we can try to give you the individual lipid concentration level in each one of those samples.
Yeah, that would be great. Thank you.
So just if you don't mind shooting me an email to remind me, and we'll copy Julian from Columbia so I can forward the data to you.
Roy, do you have the genotype differences, like for ApoE, other risk factors for neurodegenerative disease in this patient cohort?
This is going to be a question for Zeev, because this is not analysis we did, but we shared the DNA of these samples with Zeev Ganor. So I don't know if he did ApoE on them. He probably did, because ApoE can come also a neurobooster. So that will be a three-way, like we can try to obtain it from Zeev.
Yes, we do have
Thank you. Okay, great. Thank you very much.
And I think the vast majority of these samples have reached Zeev, so we can, we can potentially look at this and provide it.
Super. Thank you very much.
Sinead, I was just wondering, just technically, you have now this the NEAT method. You have like 500 plus proteins that you get. How much do you get from the depleted method? How much approximately?
So the depleted is currently ongoing. In general, we can identify around 900 to 1,000, I think, with the depleted.
Oh, right. Okay.
Yeah, you increase the number because you reduce things like albumin, these proteins that take up, are more abundant. I've yet to really dig into that data, but there's some interesting things and exciting things so far. So I want to integrate both of them together.
Interesting. Nice. Thanks. Very nice.
Okay, Julia, your slides are up.
Okay, great.
Thanks, Sinead.
What was that?
Thanks to Sinead. It's okay.
Oh, okay, great. So hi, everyone. It's nice to see you all again virtually, and hello to the new faces in the call that I haven't met yet. My name is Julia, and I'm a postdoc in the Fanning Lab. I'm excited to share with you the results that we've gotten so far studying fatty acid dysregulation in Parkinson's with GBA. To give a very brief overview of our primary focus for this work, we are doing human plasma GBA-PD fatty acid omic profiling in different plasma cohorts, where we are determining the GBA-PD fatty acidome, and this will allow us to identify novel biomarkers and therapeutic targets for GBA-PD. On the left here, you can see the discovery cohort of plasma that we have that I will be talking about our preliminary results that we've gotten so far from this cohort. And on the right, you can see the panel of saturated fatty acids, monounsaturated fatty acids, omega-6 polyunsaturated fatty acids, and omega-3 polyunsaturated fatty acids that we quantify for this profiling. This discovery cohort was obtained through a collaboration with the Karolinska Institute in Sweden. And to give an overview of the participants in it, we have 25 individuals without PD and 14 individuals with GBA-PD. For both non-PD and GBA-PD groups, we have about 60% males in each group. And the predominant GBA mutation for this cohort is the E326K mutation. In this group, the GBA-PD individuals are older than the non-PD individuals. What we found so far is that plasma fatty acid concentrations trend to be lower in males with GBA-PD. To orient you to these heat maps, what you're looking at here on the left is the fatty acid abundance of each of these fatty acids. This is calculated by the average fatty acid concentration for the non-PD individual.
Pardon me, we live in New York.
Where the lighter color represents lower abundance and the darker color represents higher abundance of that fatty acid, which is listed here. And on the right side, you see the log fold change of the concentration of each fatty acid in the GBA-PD plasma compared to the non-PD plasma. So for saturated fatty acids, we see that males with GBA-PD have a trend for lower, shorter chain saturated fatty acids compared to non-PD individuals, whereas females with GBA-PD are trending to have higher concentrations of longer chain saturated fatty acids compared to non-PD individuals. When we look at monounsaturated fats, we see overall a general reduction in monounsaturated fatty acids compared to non-PD individuals. However, the individual comparisons are not significantly different between GBA and non-GBA PD plasma here. When we look at the polyunsaturated fatty acids, we see a similar trend in lower concentrations in males with GBA-PD for omega-6 PUFAs and omega-3 PUFAs listed here. The only significant change from this analysis here is a reduction in this trans fat in GBA-PD plasma. And this is due to the small sample size of this discovery cohort. So our larger validation cohorts will be especially useful for identifying whether or not this is what we see in those bigger groups. I wanted to revisit the age difference that I mentioned briefly before because I wanted to make sure that the trends that we were seeing is not driven by the fact that the individuals with GBA-PD are older than the non-PD individuals. And so this is an aggregation of all of the fatty acid concentrations that we looked at in the heat maps before, where we see this trend for lower concentrations of fatty acids in the plasma of individuals with GBA-PD. When we look at only the non-PD individuals, we see that total fatty acid concentrations are significantly associated with a positive correlation between the concentration and increasing age, as shown here. However, in the GBA-PD individuals, this association is lost. So we found this when we looked at the significant positive correlations between different fatty acids and age for the saturated fats, where again it is lost in the GBA-PD samples, for monounsaturated fats, and also for polyunsaturated fats. So it is unlikely that the trend that we're seeing is driven by age. And again, we'll be able to look in our validation cohorts to see if we see something similar in larger sample sizes. When we look at the effects of the specific mutation that someone has, we can see that plasma C18-3 is lower in males with the L444P mutation for both the omega-6 form and the omega-3 form of this PUFA. Again, because we have a small sample size here, we will see if this replicates in our validation cohorts. But this is a promising start to see how we might be able to use this information to develop novel treatments for GBA-PD. And just to summarize the preliminary results so far, we see this trend for lower fatty acid concentrations in males with GBA-PD. We found that the age-associated changes that are normally occurring with total fatty acids over time is lost in GBA-PD. And we have identified some differences based on the GBA mutation and how this is affecting fatty acid concentrations. And this has important implications for therapeutic development. I've also looked at all of this data with the percent composition of each fatty acid. And just for the sake of brevity, I don't have time to talk about that today. Moving forward, we will be looking at the fatty acid profiles in our validation cohorts. This one is from the Harvard biomarker study, where our predominant GBA mutations are the E326K mutation and the N370S mutation. And as has been mentioned a few times already today, we also have this additional validation cohort where we will be able to determine fatty acid profiles in individuals without PD, with idiopathic PD, and with GBA PD. The predominant mutations here being the E326K mutation and the N370S mutation And we can compare this with the work that was presented for the phospholipid head groups and also the proteome. And so with that, I would just like to thank all the individuals that donated the plasma samples that make this work possible, the Silverstein Foundation and the Fanning Lab, especially Serena, and the Anne Romney Center for Neurologic Diseases, including Dennis Selko, and the Neuber Lab that we collaborate with closely. And I would be happy to answer any questions you might have.
Thank you. Thank you very much. Just more of a comment than a question, I think. It's very interesting to see that the increase in total fatty acids is lost in GBAPD. We actually observed something quite similar. The results weren't that striking, but we were looking into that, and this is also something that we are following up at the minute. But it does seem indeed that in GBAPD that there is an increase also in the GSLs, total GSLs over age, over time, but that is lost in GBAPD indeed, yes.
Oh, that's interesting. Thank you.
Yeah, thanks.
So
Hey, Judith,
I
can
was,
I ask-
sorry.
Go ahead, Judith, sorry.
I was wondering if there were collections of plasma from younger people who were asymptomatic with the GBA mutations, because I think, looking at our work in ApoE subjects, ApoE variants, there seem to be differences much earlier prior to disease onset. And I was wondering if there might be some differences earlier that we could measure and possibly mitigate before any onset of disease, or even way before something develops as a prevention treatment.
Yes, that would be so valuable to have. And I know we've talked about this with Josh in the past, but we would love to do this. We don't have those samples right now, but this is something we're really interested in doing. And I also should have mentioned that the validation cohorts that I have are age and sex match. So we'll be able to look at these profiles in individuals that are not significantly different by age, which will be really valuable.
Can I ask a quick question too? So your age effect is lost. Very interesting. Do you think there could be a interaction, not though, or in addition to be the age of onset of the disease and age, such that your interaction isn't actually lost? It's an interaction with the length of time of disease.
Yeah, I think it's definitely possible. And with the other cohorts, we have additional information like the age of onset, so we can look at that. However, it is difficult to determine exactly when the disease may have started because of the continuum of how
age of diagnosis then age.
Right. So it would be very interesting to look at, and we'll be able to see that for future cohorts. Thank you.
Thank you.
Maybe I can mention as well, I think we will be able to get at Judith's question by we have other controls that we could use. We stuck to a kind of a standard cohort that's already been published, but I think we will branch out with additional controls that we're starting to receive now. And Josh actually did say to us that A, having non-manifesting carriers will be important, and B, once we find something that we think is important, that maybe we can roll back earlier and earlier and maybe even do longitudinal studies where we have that plasma available to be able to work out how early you can identify some of these changes.
Could I just add in, that was a wonderful talk, and just to add that samples from Mount Sinai, we talked about this before, but have been submitted to BioSend. So these you can access these through the PD BRAC proposal process. It is a process, but they often grant these proposals, and we don't have as much sample because we had to put stuff into Biosend, but it's also really nice because people can pull it out. So you do have to write a proposal to PD-BRAC, to the NIH, but these samples are there and thinking about validation and so forth, but also not even validation. They'll consider a range of proposals. Sorry to interrupt. I just wanted to throw that out there as people are always looking for cohorts, and we do have some non-manifesting carriers who are in there as well.
That's great. Thank you.
Thank you. That sounds great.
Hello, can everyone hear me well? Okay, perfect. Thank you. My name is Thao, and I am from Dr. Stephanie Bennett Laboratory at the University of Ottawa. And in this project that is supported by
I
the
think you're
Silverstein
in presentation mode.
Oh, yes, for sure. Oh, I think. Do you know how to? Yeah, just a second. Good now? Okay, perfect. Sorry for that delay. So in this project, our focus is on validating the therapeutic potential of GBA1 enzyme as a transglycosidase. So this new function in the context of preventing GBA1 PD cognitive decline. Just to recap the goal of this project, we, as well as others, have identified that GBA1 enzyme has a dual functions. It can function both as a glucocerebrosidase or GCase and also as a glucocerebrotransglycosidase as a TGase. The primary function of GBA1 enzyme as a glucocerebrosidase is well known to everyone, where the enzyme remove a sugar from glucose, sphingosine or glucosoceramide to give back a glucose and a sphingosine or ceramide. The secondary function of this enzyme is as a transferase, where it can transfer the glucose moiety from a glucosoceramide and put on a cholesterol to generate glucosocholesterol. What is interesting is that to a much lesser extent, but it does exist, is that this enzyme can also transfer galactose from galactosoceramide to form galactosocholesterol. And interestingly, in pathogenic condition, the reverse reaction also happen to regenerating glucosoceramide. In the context of PD, this activity of GBA1 enzyme is much less known compared to other conditions, such as Niemann-Pick C disease, a condition which is marked by the accumulation of intralysosomal accumulation of cholesterol. The level of glucosocholesterol has been shown to be elevated. So our interest is to better understand the transferase activity of GBA1 enzyme in the context of Parkinson's disease. In a recent assignment has been focusing on how GBA1 enzyme can also go beyond just glucose and galactose and also can transfer sugar that introduced into the body from diet, such as xylose. Xylose, when introduced into the body, can form a quite rare form of glycosylated ceramide, xylosoceramide. GBA1 enzyme function as a transferase can take this xylose sugar and put on cholesterol, not just once, but multiple times to form xylose cholesterol, xylose 2 cholesterol, or xylose 3 cholesterol. All that is very interesting, and it is our focus of this Silverstein Fellowship project to fully elucidate or map out the metabolism involving the transfer activity of GBA1 enzyme. So we do this using our already established lipidomic platform within Dr. Stephanie Bennett laboratory. We extract lipid from patient sample, biofluid, namely plasma and CSF, and we use our LC-MS platform in order to fully quantify the lipid. just to kind of break this into detail, our liquid chromatography methodology allow us to separate lipids based on their different chain length, and therefore allowing for more precise quantification of lipid in the molecular species level. We recognize that the activity of GBA1 enzyme involve different activity with different sugar head group. And to separate them using liquid chromatography is challenging. So we incorporate into our pipeline differential mobility spectrometry, where we can pinpoint the level of glucosoceramide and now glucosocholesterol versus galactosoceramide and now galactosocholesterol. The usage of tandem mass spectrometry allows us to better identify each individual lipid and therefore can arrive at a precise absolute quantification. Our absolute quantification pipeline allow us to be able to report back to the clinic a level for every single lipid species in a clinical relevant manner in the level of picomole per ml of plasma of patient. Just to speak about the patient cohort that contribute to this project. We work with mainly GBA1 PD patients samples. We have 127 patients from whom we receive CSF and plasma samples. For many of these patients, we receive plasma sample and CSF samples from 1 to 7 visits. This contributes to a total of 466 samples from only the patient with GBA1 PD condition. We also include GBA1 non-manufacturing carriers in our study, and this involved 95 patients with the CSF sample and plasma sample from multiple visits as well. This contributed to 278 samples. So, and from our own bank of biosamples, we are able to provide controls to this study, 120 controls. So in total, this study is looking at around 864 samples. All plasma samples have been extracted for lipidomics and acquired by our LCSI MSMS for sphingolipid and glycosphingolipid quantification. At this very moment, post-acquisition analysis is in progress. And working in collaboration with Dr. Irina Alecu, a Silverstein Fellowship from the cohort 1, we recently have updated our differential mobility spectrometry method to make sure that we can cover the new dynamic range represented in the samples of this cohort patient. At this moment speaking, all the plasma samples are being acquired for different mobilities spectrometry. I will now present to you a new addition to this project that we currently added in. So we recognize that the activity of GBA1 enzyme as a transfer is very important. But what we're also interested in seeing is to elevating this, the measurement of this activity in such a manner that it can be done quickly, easily accessible by the clinician, and as well as by the patient. And so we have been brainstorming and working to develop a type of sensor assay, an assay where the activity of GBA1 enzyme as a transferase can be easily measured. The principle of this assay is as follows. The assay is composed of an indicator, which is a dye. It is also composed of a receptor, which has binding affinity to the dye and also to the sugar head group on our glycolipids, for example, glucose on glucosocholesterol. There is a difference in the binding affinity between the receptor and the indicator or the sugar head group on the glycolipid. And based on this differential in binding affinity, we can quantify the presence, number one, first, the presence of, for example, glucosocholesterol in patient sample. And number two is how much of this is present based on calculating the difference in the binding. Where with this idea, we are collaborating with the laboratory of Dr. Stephen Newman from the chemistry department at the University of Ottawa, who is an expert in organic synthesis and high throughput screening. So such that we can come up with a library of indicator and receptor compounds that can detect the subtlety in the different sugars attached to cholesterol, which then can be used as a readout for the activity of GBA1 enzyme as a glycosidase. And so just to summarize the progress of the project supported by the Silverstein Foundation, we focus in investigate the GCase and TCase activities of GBA1 and their contribution to GBA1 PD severity. We also ambitiously would like to map out this activity in relation to other conditions as well. We're utilizing our lipidomic pipeline with iMobility in order to perform quantification of the substrate and product involving in the activity of GBA1 enzyme. And the new development of this project, as I just showed, is our development for a rapid, easy to access colorimetric chemical assay, which can perform measurement of GBA1 transferase activity directly from a dry blood spot, which can be used in clinical for routine assessment. I would like to thank all the members in the Neurolipidomic Laboratory who have contributed to this project, notably Dr. Irina Alekiu. And I sincerely thank our collaborators who have shared with us their samples, such as Dr. Rachel Saunders-Pullman, Dr. Michael Schlossmacher, and Dr. Catherine Brookman. And thank you to Dr. Stephen Newman and Mario David Martinez from University of Ottawa for the development of the new asset. And thank you to Jonathan Silverstein for supporting this project.
Jonathan, I think you're muted if you're talking.
Oh.
Got it. Sorry about that. Let's see. Danielle, you're up.
Yes. So, is it visible?
Yeah.
Perfect. So I'm gonna update you on my ongoing efforts in elucidating the monocyte molecular and functional impairments in GBA1 associated Parkinson's disease, and as others in our foundations are. And I wanted to start with a little overview again of the reasons why we look into monocytes, the peripheral myeloid cells, circulating myeloid cells in GBA1 PD and in PD in general. Peripheral monocytes seem to contribute to disease progression in many different ways, potentially through an altered state in which they're in, which has been highlighted by now a large number of studies, including studies that came from our lab that transcriptionally characterized monocytes from idiopathic PD as well as from GBA PD and non-manifesting GBA PDs. And importantly, GBA1 function seems to be decreased in idiopathic PD as well, and it is highly expressed in immune cells like monocytes, and its activity seems to be reduced also in monocytes from sporadic or idiopathic PD cases, indicating that studying how GBA1 mutation affects monocyte function and transcriptional profile in GBA1 PD and also comparing it to other PD forms can help us find shared and distinct pathways that can be targeted in GBA1 PD and potentially in other forms of PD. And also, one important point is the fact that while many studies report an activated monocyte profile and peripheral inflammatory markers in Parkinson's disease in general, many trials that attempted to target inflammation or used immunomodulatory compounds had very modest results, very disappointing results, actually, which highlights the fact that this activation state might be just the tip of an iceberg that we need to elucidate in order to find the best way of targeting immune dysfunction in GBA1 PD and in Parkinson's in general. So that's where my effort comes in leveraging this beautiful cohort that we put up together with Dr. Rachel Saunders-Pullman from Mount Sinai Beth Israel of GBA1 PD cases, LER2 PD cases, idiopathic PD, and controls. And basically, we have monocyte samples from these donors, which we thank very much for contributing to science. And we genotype them. We do differential transcription analysis. And basically, we want to map, we want to build the multi-omic map to characterize the in-depth molecular profiles of these monocytes and compare between GBA1 PD and other PD forms and controls to understand. We basically perform single-cell RNA-seq on PBMCs, which include monocytes. We do bulk RNA-seq and bulk proteomic analysis to really map the molecular profiles in depth. And I also focus a lot of effort on a functional characterization of monocytes, of these monocytes, or more accurately, in patient monocyte-derived macrophages, which I mature in vitro from our cryopreserved PBMC cohorts. And I use the InQSight, which is an amazing apparatus for live cell imaging, to basically validate what we find at the transcriptional level, but also to really understand what the functional impairment entices here. And this may highlight concrete potential pharmacological targets to improve immune cell or myeloid cell dysfunction in PD, and in GBA1 PD in particular. Now, I wanted to dig in a bit on the transcriptional profiling we performed, which is part of our preprint. We compared the monocyte transcriptional profile from GBA1 PD patients to the sporadic PD patients' monocytes, and we found a massive downregulation in transcriptional correlates of protein translation, which is a sign of a potentially integrated stress response state. And the integrated stress response is a very conserved stress response that can be elicited by different multiple factors, including impaired proteostasis. And what the RNA-seq revealed is definitely a metabolic impairment and potential clearance issues, like from decreased lysosomal function. And down the line, we also observed downregulation in pathways related to the ubiquitin proteasome system in these monocytes. And we hypothesized basically that there may be endoplasmic reticulum and mitochondrial stress due to impaired clearance and accumulation of oxidized or misfolded proteins potentially that then causes these integrated stress response. To understand a bit better the transcriptional impairments, I created our annotated specific gene sets, because when you do gene ontology analysis or pathway enrichment analysis, the annotated genes in the different biological processes, they tend to be very broad and contain many genes. And not all of them are really truly specific to the biological process they are annotated to. So I did my own lists backed by the literature. I can also share these lists if you think it might be useful to you. It's also uploaded as a supplementary table in the preprint. And these are some of these gene sets that I compiled, and we compared their expression overall across controls, idiopathic PD, GBA1 PD, and LER2 PD monocytes. And this figure highlights how the expression of certain gene sets, for instance, the ones associated with lysosome acidification, lysosome biogenesis, and lysosomal enzymes, follows a graded downregulation across PD subtypes, with some downregulation in sporadic PD and a stronger downregulation in GBA1 PD, often also stronger than in LER2 PD. And not surprisingly, in GBA PD monocytes, we see a strong downregulation in lysosomal enzymes, or the genes coding for them. And interestingly, we also observed upregulation of genes involved in mitophagy in LER2, which was not as much in GBA1 monocytes, and genes involved in maintaining endoplasmic reticulum homeostasis were actually downregulated in GBA1 PD monocytes. And this was accompanied by a downregulation in proteasomal subunit genes and down and up regulations of different ubiquitin ligases. I found that very interesting, particularly because this is a picture that seems to be mirrored by microglia from PD cases. So recent transcriptional analysis from human postmortem tissue reveals that these transcriptional signs of integrated stress response and organelle stress, including mitochondrial and endoplasmic reticulum stress, are present also in microglia from at least sporadic PD cases. So it seems like the GBA1 mutation, and as well as LER2, intensifies this signature. And what I find very interesting in this sense is that these deregulations are very similar to what we know is going on in degenerating dopaminergic neurons, where you have loss of proteostasis, probably due to a decreased clearance capacity from lysosomal and proteasomal dysfunctions, mitochondrial stress, and this is believed to be part of the vulnerability and neurodegenerative process in these in dopaminergic neurons. And we see many similarities with what's going on in peripheral monocytes, which seems to mirror also what happens centrally with microglia. So this really highlights that there might be shared pathways that can be targeted to improve both neuronal health and immune functions. So I validated functionally these clearance pathways that I was very interested in, starting from lysosomal processing, where I used DQ-BSA, which is endocytosed by cells and then processed in the lysosomes through cathepsins, for instance. And oftentimes DQ-BSA is used in very short pulses, and fluorescence is recorded within a small time window. I did record for 24 hours because I was interested in both the initial uptake and initial processing efficiency. But then I was also interested in seeing how fluorescence decay over time, which reflects the complete lysosomal processing of DQ-BSA. And what we found is that GBA1 PD monocytes, as well as LER2 PD monocytes, they tend to retain the probe longer and have a higher rate of fluorescence emission, which basically indicates that their processing is slower. They do uptake a lot, and they have basically an exaggerated uptake, but then they cannot really process it efficiently over time. I also assessed more recently lysosome acidification through lysosensor green, and we observed decreased acidification rates in GBA1 PD monocytes. And this was also reflected in LER2 PD monocytes. So, and it's consistent with, let's say, a longer retention of DQBSA and longer rate or higher rates of fluorescence emission over time, because if the lysosomes are not properly acidified, then lysosomal processing is slowed down. And I also tested proteasomal hydrolysis through a fluorescent probe called TAS2, TAS2, and this is a non-toxic proteasomal 20S probe. It is non-fluorescent until it is actually hydrolyzed by the proteasome. And here we see we confirm that these cells, these monocytes from GBA1 PD cases, they have a slower hydrolysis. And this was true also for sporadic PD and LER2 PD monocytes. And the proteasome in immune cells is less studied, but we know it's very important for both the regulation of immune signaling and to maintain proteostasis itself. So it really seems that it's not just a transcriptional impairment in clearance, there is a functional correlate to that impairment. And this might be at the basis of this integrated stress response signaling that we see, and which may lead, to endoplasmic reticulum stress and mitochondrial dysfunctions. So these results highlight that boosting myeloid cell clearance might be a good path to reestablish immune dysfunctions in PD. And I'm also dedicating effort into better assessing this proteostasis deregulation in GBA1 PD monocytes. I did some Western blots for ISR, so integrated stress response markers and proteostasis. In the interest of time, I won't show them today. But they show upregulation of, for instance, ATF4, which is a canonical integrated stress response transcription factor involved in the regulation of ISR. And we are also about to analyze our proteomic mass spec results from GBA1 PD monocytes, and which we have for also LER2 and idiopathic PD, and we will compare the proteomic profile and see how it correlates to the transcriptional deregulation we see and integrate it also into our functional framework. And I'm also further characterizing the functional profile of these monocytes. I recently got the LysoFQ GBA1 fluorescent substrate that I want to assay in these GBA1 monocytes to monitor GCK's activity in the monocytes. So thank you very much. Thank you especially to Jonathan Silverstein and our collaborators that enables this research, and I'm open to take questions.
Okay, it seems like we can move on to the next one. We've got 4 more, I believe, and they should all be very interesting. Jade, you're up.
What happened? Can everyone see this okay?
Yes.
Okay. Hi, everyone. As before, my name is Jade. I'm a movement disorder fellow at Mount Sinai, working with Dr. Simon's and the team. Before I start, I did want to share some updates since our last meeting. So to kick off, our dual carriers study was actually accepted as a guided poster tour at the Movement Disorders Congress, which is in Houston and coming up in a few weeks. So Congress Faculty Committee will formally stop by at select posters to discuss the research. And we're pushing this project ahead with collaboration with Doctors Roy Alcalay and Avner Thaler in Tel Aviv, and others from GP2 in Colombia as well. And the second update is that our fatigue and sleep quality lighting research abstract was also submitted as a poster and accepted to the World Parkinson's Congress in May. And actually, after the last meeting, Daniele, who just presented before me, he actually shared that he was also interested in circadian rhythm. And so this organically led to a collaboration, and we submitted an FBI Accelerator Award with us and one of our lighting collaborators. So to go back to the talk today, I'll focus on longitudinal latent class analysis in genetic PD. This is a project I joined in the later phases with work done with statisticians Kathy Yang and Dr. Suiling Wang at Einstein and Sinai. This was also part of an MJF submission, and you may have seen some of this material at the GBA Montreal meeting, but I'll be presenting on some further analysis that we did. So to put our work into context, to really achieve personalized medicine in PD, we need a better understanding of its heterogeneity and factors that contribute to the disease process. And this will guide treatment, prognostication, and strengthen clinical trial design. We sought to not only better dissect heterogeneity, but to focus on latent classes to better understand how PD progresses and assess the factors or drivers associated with this heterogeneity and progression. And we took a focused look at GBA and sex-related differences specifically. So in our cohort, we looked at IPD, GBA, and LRRK2 PD variant carriers at our site, PPMI, PDBP, and the LRRK2 consortium. We used growth mixture models to look at latent classes related to motor and cognitive progression over time. So looking at MDS, UPDRS parts 2, 3, and MoCA with up to a 20-year baseline follow-up since that first visit. And in order to figure out what the optimal number of classes for our latent class study, we used the Bayesian information criterion, looking at goodness of fit, relative class size proportions, and clinical interpretability. So once we achieved this primary criterion, we then applied multinomial logistics regression models to look at which factors were associated with which latent classes. And we were particularly interested in age at diagnosis, sex, genetic variant, and hyposmic status. This is a snapshot of our entire cohort. Focusing on GBA, which composed about 400 individuals, we see that men are overrepresented, and this group had the lowest age at baseline and the shortest PD duration, but the highest proportion of these individuals were hyposmic. In our cohort overall, we looked at the group kind of prognostically without going into genetics or sex differences. We do later, and I'll show that, but this is just looking at the group overall as a whole. Our model fit indices supported a 3-class solution in slow, medium, and fast progressing classes based on the criteria that I just shared with you. When we went higher than 3 classes, the BIC got compromised and the groups became small and unstable. So that's what led to the 3-class solution. Looking at the MDS UPDRS3 progression and these latent classes, what we see is that women are less represented in the medium and fast progressing groups, but they do have a higher age at diagnosis in these groups. The slow progressing group had the longest duration of PD. And we had the highest representation of GBA in the fast progressing motor groups here. And in the slow progressing groups, you were more likely to have LRRK2. This is looking at MDS UPDRS3 progression following this multinomial regression model. And these odds ratios are associated with class membership in the medium and fast progressing groups relative to the slow progressing class. And so what we see is that older age and having a GBA variant carrier status put you at an increased risk of being part of this fast progressing motor group. Being hyposmic increased the odds of being in this medium progressing motor group. And you see that female sex and LRRK2 carrier status was actually protective against being a part of this fast progressing group. To look at MoCA progression within these latent classes, what we see is that there is a high representation of females in the slow declining cognitive class. And GBA has a very high representation in the fast declining class. What's interesting and notable is that hyposmic status becomes progressively worse as the cognitive status also becomes more severe. Looking at the multinomial regression model, but related to MoCA progression, again, having the slow progressing class as a reference, what you see is that GBA variant status and hyposmia increase your risk of being a part of this fast declining MoCA group. And female sex is protective against being a part of this fast declining group. Interestingly, LRRK2 has no effect on MoCA class membership. This is a plot, just kind of holistically looking at how GBA variant status contributes to being a part of a fast declining group across MoCA, MDS, UPDRS Part 2, and 3. And you see that GBA variant carrier status increases your risk of being in this fast progressing class across all 3 domains. Interestingly, if you're hyposmic, your risk of being a part of this fast declining group increases, but this was just the case for MoCA. In terms of what is protective, being female was protective against being a part of this fast progressing class, both with MDS UPDRS3 and MoCA. LRRK2 variant carrier status was protective against being a part of the fast progressing class with MDS UPDRS3. Looking at this forest plot, the reference group is the risk variant. And what we see is that being a severe GBA variant carrier increased your risk of being a part of this fast progressing MoCA class. Looking at the data overall with the more sex-specific lens, what stood out was that being a GBA variant carrier for men increased the likelihood of being a part of a faster progressing MoCA class. And I didn't have a chance to go into showing the trends specifically, but it was interesting that like an AD pathology with higher p-tau levels contributed to lower CSF A beta to p-tau ratios. And having this lower ratio increased your likelihood of being a part of a faster progressing MoCA class. And you saw that for both men and women, and this also held for GBA variant carriers. So in conclusion, we want to share that sex does make a difference and needs to be included in allocation schema to improve the likelihood of trial outcomes. We saw that GBA genetic status drove more severe progression in both motor and cognitive domains. And within GBA, sex and variant severity did contribute to heterogeneity in both motor and cognitive progression. And I did also highlight that cognition was at first adversely affected in GBA men. Olfactory phenotype is less helpful in GBA PD, plays a greater role in LRRK2 PD. And as far as implications of for our study, what we posit is that there may be an alternative mechanism of cognitive decline in women with GBA PD that should be considered. Our study may also inform sex-related differences and mechanisms of PD progression with a focus on tau in female subgroups. And we should also consider sex-related stratification in clinical trials especially with cognitive outcomes. So thank you for your attention, and thank you for the Silverstein Fellowship for supporting our research.
Okay. Any questions for Jade? Okay. Georgia, I believe you're up. Thanks, Jade.
Okay. And can you please confirm that you can see the slides?
Yeah, perfectly.
Perfect. So today I will present to you updates in my project that focuses on investigating the commander complex as a modulator of lysosomal function in neurons from GBA1 PD. So the commander complex, as a short background intro, is a 16 protein complex that comprises of protein ring COMD1 to COMD10. These come together with cold coil domain containing protein CCDC22 and 93, and this allows to bring together the retriever that and then 10, therefore comprising the full commander complex. And what this complex does is that it comes in contact with specific adapters. The one that is currently studied is sortinexin 17 and participates in the trafficking from the early endosome back to the plasma membrane. Now that was its known function. And last year we found that actually a commander complex is also important for lysosomal glucocerebrosidase activity. Deficiency leads to a drop of GCase activity, but also general lysosomal proteolysis is deficient. And importantly, we have seen that commander complex deficiency generally affected the trafficking of certain lysosomal proteins. They cannot make it to the lysosome. And some of those proteins are released from the cell in association with exosomes and extracellular vesicles. Importantly, genetic analysis showed that loss of function variants in commander complex may increase PD risk. And therefore, in this project, we're mainly trying to understand what is the mechanism by which commander affects lysosomes and how does this modulate the GCase-related phenotypes in neurons from GBAPT individuals. So for today's experiments, I'm mostly, again, investigating how does commander impact lysosomes. And to remind you, what we had shown is that commander deficiency leads to within endolysosomes. So these are enriched fractions from cells stably expressing LAMP1 RFP3XHA. Endolysosomes are enriched by magnetic isolation, and these fractions seem to have less GCase, less LIMP2, and some other proteins. Moreover, some proteins were higher, and this included prosaposin and progranulin. We found, interestingly, that not just their processing was deficient, mostly their very robust phenotype is that the full-length precursors seem to accumulate within these endolysosomes by 3 to 4-fold. So as part of AM1, we try to understand how that works. And part of it that I'm focusing today is the performance of a proteomic analysis using tandem mass spectrometry to understand global protein changes in early endosomes and later endosomal fractions. And what we're hoping to gain out of it is to potentially identify not just generally what's wrong, but some specific vesicular adapters that may be dysregulated and hint to us to what does commander does not come in contact to, thus leading to mistrafficking of GCase, LIMP2, and other proteins. So here you can see the expression of our of our constructed TMM192 within the cells. We also have LAMP1, and we think these are enriching the endolysosomes, so late endosomes and lysosomes. And we see these constructs are not really overlapping with EA1 domains. So theoretically, this means that we should be able to differentiate between early endosomes and late endosomes and see whether any proteins are being stalled in these compartments. So in the lab, we had all these protocols previously established, but scaling this up for proteomics actually became much more challenging than I originally estimated. And I'm presenting to you now the optimized protocol, which actually did take me 3 months to manage. So initially, we start with two 15 cm dishes of comfortable confluence. The cells are expressing either of these three constructs individually, and then the clear supernatant is mixed with a set volume of magnetic beads. This step had to be optimized. Then after incubation, this is washed, and that actually makes a difference in the final yield. And ultimately, we elute in a set volume that had to be optimized in a buffer compatible for proteomic analysis, because what we want to present to the core is ideally 100 micrograms. 25 microgram could also work for this analysis, but the truth is that the higher amount you have, the more you could detect certain peptides and, not miss some existing differences. So what we first asked is what yield do we get amongst these fractions? And each experiment was performed in duplicate. So I'll just show you what I have gathered today. I see that it is feasible to get, let's say, 30 to 50 microgram per fraction, which means that pooled, it can yield from 70 to 100 microgram. That could be good to submit for proteomics. Then for the TMM192 construct, I seem to have a little bit more variability. So that still needs some maybe further upscaling, maybe use more cells to begin with. And last, the late endosomal fractions. Here, as much as you try with these conditions, you get about 15, so basically 30 microgram per preparation. So I simply think I need to double this amount. And that's what I'm working on now to finish all these fractions. But now there was another interesting question for me was, so one question is to find adapter proteins that can explain the impact of commander complex. Another one is, could this experimental setup answer to me whether certain lysosomal proteins are stalled in early endosomes or late endosomes or lysosomes? So for that, I asked whether by putting both EA1 and LAMP1 fractions on a blot, how well are these fractions really separated? So, so what you see here is input versus IP. This part is for the EA1 blot, and then the other part is for the LAMP1 IP blot, and again, it's input and IP. So what do we really see? So initially, we see certain markers of early endosomes that there could be a little bit of overlap between membrane proteins associating with early endosomes. For example, RAB5 is also present in the late endosome fractions. Then we have LARP1. This is a plasma membrane protein recycled via the early endosome. And you can see here it is present both in the early endosomal and late endosomal fractions. But let's say this could be explained because it goes there for the gradation. But then we also saw a very good enrichment in the later endosomal fractions for LAMP2 and especially LIMP2 and some luminal proteins, including HEXP, catepsin B, and I can tell you the same is the case for GCase, progranulin, and prosaposin. So basically, what does this tell us? It says that it could be either technical or biological, that there is a little bit of overlap especially in the membrane proteins participate, like that are present in these fractions, but the luminal proteins actually show a good separation, so it could be reliable to make conclusion based on the relative abundance of a lysosomal protein in either fraction. Another thing that was important is that we also looked at the association of commander complex components COMD3 and COMD1 with both fractions. And interestingly, we saw that COMD1, which is again part of the commander ring, is associating also with the late endosomal fractions, which I think there is two alternatives. Either it will be ultimately dismissed as a technical finding, or in fact, and I think that would be the more interesting case scenario, there could be other adapters that the commander is coming into contact that are present in subtypes of late endosomes and therefore can modulate the trafficking of certain lysosomal proteins. So in conclusion, we are ready to submit our fractions of LAMP1 positive for quantitative proteomics. I'm currently upscaling a little bit for the early endosome and TMEM 192 fractions. I think there is a good degree of differentiation between the proteins encountered in early and later endosomes and lysosomes. And another thing is for the evaluation of the results, I'm always looking at what controls can I use, and I think there are some reliable internal controls that will help to evaluate how important is a difference in other proteins. This includes number one, the depletion of LARP1 as a canonical substrate of the commander complex in early endosomes. Moreover, the depletion of COMD3 and COMD1, which can be expected because this destabilizes the entire complex. And B, another thing that's extremely robust and reliable is that we have seen this 3 to 4 times accumulation of full-length progranulin and prosaposin in the lyso fractions. We don't know why, but it's happening very much. So I think these could be some standards to evaluate how important other differences are. And ultimately, we think that as we are moving towards other experiments with neurons, the answers we will get from this type of experiment will be important for revealing some other molecular players that associate with the commander and contribute to mistrafficking of lysosomal proteins, and potentially provide some new key insight into how commander deficiency affects lysosomal function, which is a key, and the dysfunction is a key pathomechanism is GBA1-PD. So ongoing is, the finalizing for the next 3 months of all these fractions to be submitted from proteomics, currently expanding eye neurons and dopaminergic neurons to validate some of the things within the next 3 months, and also examination of where exactly GCase goes under commander dysfunction, because we know less of it is in the lysosomes, but is not identified in exosomes. So we are suspecting maybe some issues related to lysosomal exocytosis. This experiment could not be done so far in HEC cells because it's just not detectable, but I think maybe in neuronal settings that the medium is conditioned for longer, we will have some clear answers. And I thank you all very much, and thank you very much for the Silverstein Foundation for supporting the research.
Okay, any questions, Georgia? Okay, Jeff, you're up.
All right. Can everyone see?
Yeah, perfect.
All right, great. I know we're quite behind, so I'll try to keep this rather brisk. So just a quick background. We're all familiar with lysosomal storage disorders, and they have been associated with Parkinson's disease risk. And in a previous fellow from Baylor, she found that there's a synergistic effect between GBA and ATP13A2, which can cause Parkinson's disease in an autosomal recessive manner. So we figured that there are likely other human cases where we may see a synergistic effect between GBA and other lysosomal storage disorder genes. And in fact, we believe that GBA-PD has a meaningfully different lysosomal genetic architecture compared to other PD. So instead of doing a typical case control analysis, we performed association tests between GBA-PD and non-GBA-PD. And as an update for this meeting, we have incorporated now African and Ashkenazi Jewish as well as UK Biobank dataset, which more than doubled our dataset. So all those results you see here is a result of the extra sample sizes, and it's basically new information at this point. So the design was to first extract pathogenic or predicted loss of function variants in these lysosomal storage disorders, compare the level of burden between GBA carriers and non-carrying PDs. And then after that, we try to identify subpathways within the lysosomal storage disorders or genes that are enriched or depleted in GBA-PD compared to other Parkinson's disease patients. And first, when we look at across all lysosomal storage disorders, we see a very, very strong signal of 10 to the negative 5. And just as we hypothesized, we found that there is a big difference in lysosomal burden between GBA-PD and other PDs. And when we look at subpathways within the lysosomal storage disorders, we see glycosphingolipid catabolism genes as well as mucopolysaccharidosis genes. Neither of them are a big surprise. Glycosphingolipid catabolism is, of course, the sphingolipid pathway that GBA is implicated in, and the MPS has already been established in a previous literature. But this shows that this is the first, what we're doing seems to be right. It's unsurprising that GBA interacts with other genes in the pathway, and we are able to confirm previous literature. Now, what's interesting is that when we break down the glycosphingolipid catabolism even further, to upstream of GBA versus downstream of GBA, we see the opposite effect. So if we think about it in a way, if we look at the upstream of GBA, and if we think that those lose function, we will likely get less lactosoceramide and glucosoceramide, which are the substrate of GBA. So as if almost like a natural substrate reduction therapy. And we see that it's potentially we see a potential protective mechanism. On the other hand, downstream effect, we see a greater synergistic effect. So the product, the ceramide, would be increased. It would be decreased in the case of a double knockout of GBA and say SMPD1 or ASAH1. So it is really cool to see that we see this contrasting effect, and we can see within the pathway, we can see how human genetics can reflect what we already know about the lysosomal pathways. So after that, we decided to look at individual genes rather than looking at pathways. And what was really interesting is that the top 3 genes are not directly related to a glycosphingolipid pathway. In fact, the very top one is a lysosomal cholesterol transporter, as well as an MPS gene and SLC17A5. Now, GAL-NS is a MPS gene that, as I mentioned before, has been implicated in a previous literature, and SLC17A5 was implicated in our lab's previous paper that found an association with just general PD. But to our understanding, NPC2 has never been associated with PD in the scientific literature, and when we do our own case control analysis, we get a null signal. And we also performed a replication analysis in a completely separate independent GP2 data that included genotyping data as well as clinical exomes, and we were able to replicate this association for GBA-PD specificity. So of course, GBA is multi-allelic, right? Different variants cause different level of risk, and we know that there are some functional differences between the variants. So we thought, hey, let's look at these effects when we isolate them by alleles. And what we see is that NPC2 interaction is very specifically T48M, or also known as T369M, and N409S carriers or N370S. By contrast, E326K or E326K didn't show any significance at all. And again, this was replicated in an independent data, and once we meta-analyze it, we can see that T48M and N49S has a very, very strong significance with -6. Meanwhile, E326K almost shows a null signal here. So this definitely requires some investigation in the future. So we found this loss of function as function evidence in the human genetic data, and we thought of other ways we can try to better understand the interaction between MPC2 and GBA. So we integrated UK Biobank proteomic data to perform what's called a Mendelian randomization, and it is basically using genetic data as well as proteomic data to try to test for the causal association of exposure to outcome. In this case, our exposure was MPC2 levels in the blood, and our outcome will be GBA-PD, specifically the carriers of the aforementioned variants. And again, we see the result where reduced MPC2 protein increases the risk for specifically GBA-PD. And once we try to interpret the outcome, it shows that those carriers at the lower end of the MPC2 levels have almost double the PD risk compared to people who have normal levels of MPC2. So our next step is to look at other lysosomal genes. So for example, we're planning to do a burden analysis of Commanda complex. As Georgia mentioned before, Commanda complex showed that it can increase PD risk. But they haven't been able to test GBA-PD specifically due to sample sizes. So we're hoping that we can use this multi-ancestry sample to have a better power to see if we can see a interaction between GBA and the Commanda complex. And we're also looking at the potential clinical descriptions of these double carriers of GBA and MPC2, and preliminary results are showing earlier onset and higher baseline levodopa requirement. And there are a few thoughts to chew on that's really interesting is there's multiple evidence in the literature now that shows that E326K is kind of unique, whether it be lipid droplets or the result from Denali that's showing that E326K has changes in binding with LIMP2. And this is another evidence to show that maybe E326K really is unique and confers risk to Parkinson's disease outside of typical GCase activity. And of course, the role of cholesterol trafficking in GBA-PD, as mentioned by Dr. Bennett, and a presentation by Tao, is really, really understudied. And we hope to potentially collaborate with them to better understand the role of cholesterol trafficking and potentially interaction with GBA and MPC2. And I would like to thank my lab as well as the people who donated their samples to GP2 and UK Biobank because they make the research possible. And of course, to Jonathan and Silverstein Foundation for their support as well. Let me know if you have any questions.
Super interesting work. Just to comment at the looking at the at protective variances, I think super interesting. I think Ollie Isaacson had a grant from the Fox Foundation to looking at hexominidase A, and there was also an older paper where I'm not sure if it was a British paper, but I don't recall if it was hypothesized or if they had autopsy data to support that basically just being heterozygous to Tay-Sachs is quite common in Ashkenazi Jews if that's protective. And I don't think we've seen any signal with that, but super interesting.
Yeah, that's it. Yeah, like when that first came up, I think while I was doing the literature search saw some the stuff you mentioned before. Yeah, and this would be like almost like a natural substrate reduction therapy, but like given that Venglustat didn't really work out, I'm not sure what to really make of it.
I have a comment, question?
Yep.
Hey Jeff, good, nice presentation.
Thank you.
I think we should contact offline about the commander burden, because we currently have a project on GB2 with Nicola Mencaci and expanding the analysis of the entire the commander complex in GP2 families, and also seeing exactly what you said, that the interaction with GBA alleles that we weren't able to do for the paper because we didn't have the GP2 access. So would be good if we could brainstorm on how to move forward with that, because that's very interesting.
Yeah, let's definitely contact after this meeting about it.
Sounds good. Thank you.
All right, Andrew. Are you there? Okay, we're ready to go.
Okay. Thank you for your attendance and the last one of this present update meeting. So hello everyone, I'm Andrew Zhu from Professor Monza Abreu-Malis lab at Stanford University. So today I'm going to briefly update the recent progress integrating the cellular and lysosomal lipid profiling of GBA-PD patients to identify the clinical biomarkers. So in collaboration with clinical researcher, Dr. Esther Samler at University of Dundee at UK, we were able to recruit GBA1 variant carriers with PD, including the T408M, E365K, and also some L483P patients, those variant carriers, and also some healthy controls. So we collected the specimens, including urine, blood, and also plasma. And we were able to isolate the monocytes and also PBMCs, the peripheral blood mononuclear cells from the peripheral blood. So we were able to then extract the molecular contents, including the proteins, lipids, and also metabolites from those cells. And we are also able to do the multi-omics analysis at whole cell level, and also the lysosomal levels using the tagless lyso-IP. So we've done the untargeted lipidomics analysis of the PBMCs. So our lab have been developed techniques called lysosomal immunoprecipitation, lyso-IP. So to investigate molecular contents in the lysosome. So previously, we engineered the lysosomal membrane protein TMEM192 with 3 times hemagglutinin tag on the cytosolic side. So with the anti-HA antibody conjugated to magnetic beads, we were able to isolate and enrich those intact lysosomes from the cells and also mouse models after the genetic engineering. But that's not applicable to the clinical samples because we cannot do the genetic engineering for those patients. So that's why we recently developed the tagless lyso-IP that we use antibody that can recognize specifically to the endogenous TMEM192, the lysosomal membrane proteins. That antibody is conjugated to magnetic beads, so we can directly enrich the endogenous lysosomes from the clinical samples, including any human cells and also tissues. So basically, this following the same lyso-IP workflow that can do the immunoprecipitation in less than 10 minutes, that are able to that allow us to analyze the molecular contents in the clinical lysosomes and also for further immunoblotting and also enzymatic activity investigations. We also implicated that tagless lyso-IP to human brain tissues, and we were able to see the pretty good over 6-fold enrichment of the specific lysosomal lipids, BMP lipids. And we were also to enrich some lysosomal proteins using those lyso-IP method. So from the lipidomics data of whole cell level and also lysosomal levels using the Taglis IP, we performed the PCA analysis of the PBMC PBMC cells from the GBA1 variants and healthy controls. So from the whole cell level, from the positive mode data acquisition, also negative mode data acquisition, which is the top two PCA plot. We didn't see much differentiation of those PD patients and also healthy controls. Looking at those lyso-IP PCA analysis, we still didn't see much separation in positive mode data acquisition. Although we see some separation in negative mode, but the healthy controls are still like mixed with the PD patients. So which means those linear PCA analysis may not super clearly separate the PD groups from the healthy controls. And we are going to test the non-linear embeddings and spectral clustering to facilitate better separation of those patients. So we also investigated the sex differences in GBA1 variants. For example, in the E365K carriers. So from the whole cell level on the left, we investigated patient and healthy controls from both sexes, but we didn't see much significant lipids there. On the right is actually the Taglis lyso-IP data, also of the two sexes combined. We still don't see much significant lipid changes. But when we look at the male carriers only, we see a very significant downregulation of those phosphatidyl inositols, PIs, that's showed up on the top left. That is related to the downregulation in the E365K male carriers. And for the female carriers on the right, from whole cell level lipidomics analysis, we were able to see the upregulation of some hexaceramide showing in green dots. That's on the top right of this volcano plot that's showing good upregulation as expected in those GBA1 female carriers. So these are pretty preliminary data. We are going to keep analyze other GBA1 variant carriers. So for the next step, we are going to keep performing the lipidomics of those monocyte samples. And we are going to optimize the nonlinear embeddings and also spectral clustering to improve the separation of the GBA variants, carriers, and also the healthy controls. And for the lipid species with significant changes, we are going to perform targeted lipidomics using the triple quad mass spec to quantitatively validate those lipid species alterations. And in the end, I want to thank the Silverstein Foundations for the support and also Abrimalis lab members for the help and also Semler's lab for recruiting those patients and collecting those samples and also those patients and healthy volunteers who participated in this study and also the funding support. And thank you all for listening and good suggestions.
Okay, great. Any questions? Okay, just 30 seconds here, but May 21st is our next meeting. It's an in-person meeting. We're doing one in-person meeting and 3 Zoom calls in a year. And so this is in Phoenix, Arizona, where the World Parkinson's Conference is. And it will be, it is, I think it was stated just a few minutes ago. It's going to be back to back with the GBA1 conference and our conference. So you can fly in on them Monday and be there for as many as 6 days if you wanted to live at conferences. So please join. Danielle's going to send out everyone else's contact information. If your contact information has changed, please let us know at info@silversteinfoundation.org. And otherwise, we'll get the information out. Several people have requested the list again, so we'll get that to you. Anyway, great meeting. Really proud of what you guys have accomplished, and can't wait to see what's next. So, thank you again, and we'll talk soon. Bye-bye.
Thank you, everyone.
Bye.
Thanks.
Bye, everyone.
Thank you. Bye.
Thank you.